Hello.
Hello. Hello.
हाँ साहेब.
Okay. मस्के साहेब. Hello. मस्के साहेब. तुमचं हे माईक हे करा.
हॅलो.
गुड मॉर्निंग साहेब. नमस्कार.
गुड मॉर्निंग सर.
ओके, सुरू करतोय.
जॉईन होतोय मी.
हो. ओके.
Hello. Good morning, everyone.
काकांचं झालं का?
नाही होतंय, तुमच्या नंतर होईल ते
माझ्या
ऑनलाईन.
काका जॉईन होतात का कसे?
त्यांनी मेसेज पाठवलाय, ऑनलाईन आहे तो. होईल ते 2-3 मिनिटांमध्ये.
हो का?
हो. अडचण.
मग काकांचा मेसेज झालं की मग आपल्याला सुरू करता येईल.
Hello. Good
हो
morning,
हो, मी
everyone.
आता सुरू करतोय. हॅलो.
हॅलो.
हा, सुरू करतोय आपण आता एक थोडक्यात.
हो, मग तुम्ही सांगा मला जेव्हा हे करायचं तेव्हा.
नाही, आता सुरू झालंय. आपण एक थोड्या वेळातच आपण पूर्ण करूया, अर्ध्या तास.
पहिल्या सेशनला वक्ते कोण आहे?
तुम्ही पहिलं हे करा, इनॉग्रेट करा, आणि त्याच्यानंतर कीनोट ऍड्रेस जे आहेत आपल्या, त्या मॅडम आहेत
बर बर बर.
बँकेच्या, त्यांचा कीनोट ऍड्रेस आहे.
कीनोट ऍड्रेस आहे त्यांचं.
पहिलं तुमचं हे इनॉग्रेट करून घ्या.
बरं.
हो मॅडम, कंटिन्यू. हॅलो.
हॅलो. सुरू करा.
A very good morning to everyone. Join over here. I take this privilege to warmly welcome to all the participants of online international conference on advances in life sciences, biodiversity, and conservation. It's our immense pleasure that today's international conference is jointly organized by Department of Botany and Zoology under the aegis of Internal Quality Assurance Cell and our college. On the behalf of Sri Sadguru Gangagir Maharaj Science, Gautam Arts, and Sanjeevani Commerce College, Kopargaon, I extend my deepest appreciation to all of the esteemed speakers, distinguished guests, and dedicated participants for the gracing this occasion with their presence. It is a privilege to assemble such a gathering of intellectual minds as we embark on an enriching exploration of breakthroughs in life sciences, biodiversity, and conservation. Science is not merely a pursuit of reason, but a journey of curiosity, passion, and discovery. And today, we unite in that shared quest for knowledge and innovation. I want to extend my sincere gratitude towards respect.
हॅलो.
हॅलो.
सर सर, आवाज जातोय का मधून मध्ये?
हा, थोडस टेक्निकल.
Am I audible?
येस.
करा, कंटिन्यू करा. करा मॅडम, कंटिन्यू. आवाज गेला होता.
I want to extend my sincere gratitude towards respected Honorable Principal Professor Madhav Sarade sir for providing this opportunity to our Botany and Zoology departments to host today's international conference. For that, first of all, with immense respect and admiration, I would like to welcome a visionary leader and guiding force, Dr. Madhav Sarade Sahib, Principal of Sri Sadguru Gangagir Maharaj Science, Gautam Arts, and Sanjeevani Commerce College, Kopargaon. Then, I am delighted to extend a warm welcome to today's inaugural eminent speakers and resource persons, organizing committee members, Vice Principal of the college, NAC Coordinator and Convener of the today's conference, Dr. Nilesh Malpuri sir, Organizing Secretary Dr. Jiv Tode sir, HOD of Zoology Department, Coordinator Professor Mate sir, HOD of Botany Department, and all respected teachers, members, and my dear participants. Welcome one and all. Now, I would like request our Honorable Principal Professor Dr. Madhav Saradi sir to grace the introduction of today's online international conference. Welcome, sir.
Thank you, madam. Good morning to one and all. Firstly, I would like to extend my humble regards to Padma Bhushan Dr. Karmil Bhaurao Patil, founder of Prayas Shikshan Sanstha. Myself, Principal Dr. Madhav Sarode, SCGM College, stand before you today to deliver the welcome and introduction of inauguration ceremony at this remarkable international e-conference. It gives us immense pleasure to welcome you all on behalf of Sri Sadguru Gangagir Maharaj Science, Gautam Arts and Sanjeevani Commerce College, Kopargaon, Maharashtra, India, organized by Department of Botany, Department of Zoology and IPSC on advances in life sciences, biodiversity and conservation. Today's on this occasion, our President, Honorable Advocate Bhagirath Sinde, aside, Vice Chair Maharaj Shikshan Samstha Satara, Chairman of the CDC, SGM College, Kopargaon, and today's our chief guest for this inaugural functions, Honorable Dr. Naneshwar Maske Sahib, Vice Chancellor, Karmil Bhaurao Patil University, Satara. Also, on this today, overall, our plenary speakers, keynote speakers, Honorable Dr. Deepthi Ekandwala, Madam, Senior Professor and Head, Department of Botany, University of Peradeniya, Sri Lanka. Also, the plenary speakers, Honorable Dr. S.R. Yadav, Sir, Indian National Science Academy, Honorary Scientist, Department of Botany, Shivaji University, Kolhapur. Also, Honorable Dr. Shakti Kumar Singh, Scientist D, Ministry of Environment, Forest and Climate Change, New Delhi. Also, Honorable Dr. Leti Alesh Sinnu, Scientist C, Central University of Kerala. And also our Honorable Dr. C.G. Khilare, Sahib, Principal, Sharat Chandra Pawar Mahavidyalaya, Lonan. A warm welcome. I extend warm welcome to researchers and participants from around the world, our patrons, advisor committee members, organizing committee members, and dear students. At the outset, I take immense pride in introducing the Raya Shikshan Samstha, an institution that has been the beacon of education for over a century, founded by Padma Bhushan Dr. Bhaurao Patil, with the noble vision of bringing education to the masses. The Samstha has grown into one of Asia's largest educational institutions with an expansive network spanning multiple disciplines and regions with 752 branches, 43 colleges. Our mission has always been to educate, enlighten, and empower. This conference serves as a dynamic platform for researchers, academicians, environmentalists, and policymakers from across the globe to come together and exchange innovative ideas, latest research findings, and future direction in the fields of life sciences and biodiversity conservation. In the face of rapidly changing environmental conditions and increasing threats to natural ecosystems, the importance of interdisciplinary approaches in life sciences has never been more critical. This conference aims to highlight cutting edge advancements in areas such as biotechnology, ecology, environmental sciences, and genetic research, while also addressing pressing issues related to the biodiversity loss, climate change, and sustainable development. By fostering meaningful dialogue and collaboration, we hope this event inspires actionable solutions and strengthens global efforts towards preserving the rich biological diversity of our planet for future generations. Over the day, we look forward to provocative discussions, groundbreaking research presentations, and collaborative initiatives that will help shape a more sustainable and resilient future. Let us make the most of this platform to inspire, connect, and contribute meaningfully to the global scientific community. Thank you. And once again, a very warm welcome to all dignitaries, distinguished keynote speakers, esteemed guests, panelists, and participants and dear students. Let us make this conference a memorable and impactful one. Thank you. Thank you. Over to Lavanya madam.
Thank you so much, sir. Before moving ahead, a kind request for all the participants. Please switch on your device camera and mute your mic for a minute to take the photograph. Okay, thank you. Now, let's move on. Online conference presidential address. For that, I would like request to Professor Viral sir from technical session to start the AI speech of President for the inaugural function. Honorable Advocate Bhagirath Shinde Kaka, Vice Chairman of Rayat Shikshan Sanstha Satara.
Good morning everyone. Myself Advocate Bhagirath Shinde, Vice Chairman, Rayat Shikshan Sanstha, Satara and Chairman of College Development Committee, would like to express my views on this international e-conference jointly organized by Department of Botany, Zoology and ICAC. The Rayat Shikshan Sanstha, Satara has been contributing immensely to educate the masses in the Maharashtra through 752 branches. The network of its branches has spread like branches of the banyan tree. It is spread over 14 districts of Maharashtra and one of Karnataka with 14,990 employees and 4,59,729 students belonging to all religions, castes and creeds. The present international e-conference is one of the efforts to attract the attention of the academic arena towards a very vital topic, that is biodiversity, which ensures a resource for new food crops and medicines. It is facing threats from increasing population, pollution, deforestation, and species extinction. Extinction is progressing at an alarming rate. This in turn affects other dependent species and altering the ecological balance. Conservation of biodiversity in natural habitats including protected areas, sacred groves and other biodiversity hotspots is crucial for maintaining the resilience of ecosystems. The International E-Conference on Advances in Life Sciences, Biodiversity and Conservation gathers leading experts, researchers and professionals from botany and zoology. This e-conference aims to foster collaboration and exchange of ideas to drive innovation in the field. Participants will have the opportunity to learn about the latest advancement and trends in life sciences through the renowned experts such as respected Vice Chancellor Honorable Professor Dr. D.K. Mhaske, Karmveer Bhaurao Patil University, Satara, Dr. Deepthi Yakandwala, Senior Professor, University of Perdenia, Sri Lanka, Dr. R. Yadav, Eminent Taxonomist of India, and all distinguished resource persons and participants. On behalf of Rayat Shikshan Sanstha Satara, I would like to welcome you all in this wonderful international e-conference. Overall, the e-conference is a platform to stay updated on the recent trends and innovations and drive positive changes and collaborate with peers and academicians, the scholars across the globe. SSGM College Kopargao has organized 02 international and 03 national conferences since February 2025. I am very much happy to inform you all that this e-conference is adding another feather in the research contributions by the college. I wish all the best for the efforts taken by Principal Dr. Madhav Sarode and his team, and once again I welcome you all in this wonderful international e-conference and also congratulate to you for organizing such a beautiful international e-conference. Thank you. Thank you so much to one and all.
Okay, thank you so much, sir. It's time to inaugural functions. So that it is with immense pride and great pleasure that I introduce our distinguished guest, Principal Dr. Nandu Pundlik Maske sir, the Vice Chancellor of Karmveer Bhaurao Patil University, Satara. Dr. Maske sir is highly respected educationalist, researcher, scholar, administrator, amateur bird watcher, and environmentalist with an illustrious career spanning over 36 years in the field of higher education. He holds an MSc and PhD in zoology and has served as lecturer for 16 years and principal for 20 years. Since 14th June 2024, Sir has been serving as the Vice Chancellor of this distinct university. Throughout his academic journey, he has significantly contributed to educational development at RBNB College Sirampur and MJS College Srigonda. Sir has initiated 2 undergraduate and 8 postgraduate programs and 4 research centers, then successfully completed 2 major and 2 minor research projects, and authored 8 books and edited 5 books, and published 39 research papers in national and international journals of repute. Sir has represented his expertise at over 32 international conferences, seminars, and workshops, and has also organized 22 national level academic events. Sir has played an active role in Science Association and the Avishkar Initiative, further promoting research culture among students and faculty. Sir served as district coordinator of NS units in Ahmednagar district, and under his leadership, the unit received the prestigious Nirmal Gram Puraskar from the President of India. Sir's social commitment is also reflected in his work with Anandavan for leprosy eradication and rehabilitation, and his support for the Pasipardi tribal community. As the part of national mission, he developed a biodiversity register for Srigonda Tehsil and has been a key figure in spreading awareness about integrated pest management. Dr. Maski sir has also conducted 2 faculty development programs in association with MSFDA. Sir was instrumental in establishing a Tata BPO call center at RBNB College, Sirampur, under aegis of Tata Business Support Services, promoting skill development and employability among rural youth. He is a life member of the Bombay Natural History Society and actively works with international organizations UK and Royal Society for the Protection of Birds UK. Sir shares strong academic ties with global educators, including Professor Louis Mike from Salisbury University, USA, Mr. C. Dale McDonald from Denver, USA, and T. Mong Mayak University, South Korea. Dr. Maskar sir is also research guide under Savitribai Phule Pune University, mentoring 6 PhD students, and has served in multiple academic bodies, including Chairman of the Board of Studies in Zoology, Academic Council, Faculty BUTI, Grievance Committee, and as an assessor of NAC for 5 colleges. Sir is the Joint Secretary of Rayat Shikshan Samstha Satara and a member of its managing and executive councils. His institutions have received honor such as the Best College Award at 2007 and the Best Principal Award at 2008 from Savitribai Phule Pune University, Pune. Dear participants, Dr. Maski sir is a truly multifaceted personality and a passionate academician, a nature enthusiastic, and a leader deeply committed to social development, whose remarkable contributions in the field of life sciences have set benchmarks for all of us. Let us all extend a very warm welcome to Principal Dr. D.K. Maske sir. I heartily welcome you, sir. I would like again request to sir to share the thoughts with us for inaugural functions.
Good morning.
Good morning,
morning.
sir.
Good morning to one and all. Principal Dr. Madhu Sarode, Principal of the SSGM College, Kopargaon. My beloved friends, Professor Malpure, Professor Jutode, Professor, actually very distinguished speakers. Professor Yadav sir is also seen to have joined the inaugural function. All distinguished guests from across the country and from abroad. I'm very happy to be a part and parcel of this inaugural function that SSGM College has organized a conference on advances in life sciences, biodiversity, and conservation. Actually, about hello,
Hello, sir.
Yes, sir. येतोय आवाज.
is my voice audible?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes,
You are
yes.
audible, sir.
About 8.7 million species of plants, animals, and microbes have identified as to it. Of these different species, 6.5 million species of plants and animals are found on land, and nearly 2.2 million species have been identified from the oceans. India itself has a 45,000 plant species and somewhere around 91,000 animal species. Across the world, 17 mega biodiversity hotspots are identified, and of these 34 biodiversity hotspots, India alone has a 4 global biodiversity hotspots. Now it's time advancements in various sciences and technologies. The traditional view of conserving and managing biodiversity is seen to have been changing very fastly. Basic advances that we have that can help to conserve biodiversity, make a management of biodiversity are like this introduction of artificial intelligence, machine learning, and computer vision can make a drastic change in our traditional way of thinking of biodiversity management. Basically, with the help of this artificial intelligence and machine learning, we can analyze large database. Then we can have information about global biodiversity. We can predict the species distribution, and we can identify the threat to biodiversity. One more advances in life sciences that we can have with this computer science is eDNA, environmental DNA, and genomics. Earlier, as to have a knowledge as to what species are there in a specific habitat, we have to observe them on the land itself or in the habitat itself. But now, with the help of this artificial intelligence and machine learning, we can have an indirect method of identifying which different plants with different animals might have been there, that in the habitat, with sampling the soils, waters, and these techniques can help us to identify species without directly observing the species. And this genomic helps us to understand the genetic diversity and adaptation thereon in the field. Then network sensors can also help we people as to monitor various environmental parameters that may help us to provide real-time data for bioconservation and management. Besides, we can have a different techniques like this genomic, then network sensors, then global navigation satellite systems, and we can have different ideas with these techniques as to how we can conserve this biodiversity, may it be a plant or animals. And for conserving the biodiversity, we need to take care of it that with this AI and machine learning, we can minimize the cost of this conservation planning. Besides, we can restore the degraded ecosystem. We can have an insight in protected areas and habitat management. We can establish and protect a habitats with effective management system. We can have a species recovery, and this endangered and threatened species can be neatly managed. Besides, we can have a basic ecosystem services like clean water, pollination, climate regulation, and all these things. These new advancements in science and technology, especially this computer science, can make us to help this biodiversity conservation and biodiversity management. We can restore it, and we can manage it. And for this conservation management, we can use new tools like this camera tapping, then bioacoustics, biotelemetry, brain mapping, remote sensing, airborne remote sensing, Google Earth engines, then this unmanned aircraft systems, GPS telemetry, wireless sensors, biometric devices, and we can have a neat data analysis with the help of this artificial intelligence. Besides, as to make this biodiversity conservation and management, we need to have some planning, we need to have strategies and policy making to conserve the biodiversity. For that, we can have this environmental change, ecosystem natural appropriate acts for this management and conservation. For conserving the biodiversity, we need to have policies for this climate engineering, cloud seeding, this desalination of seawater, then geoengineering, cryogenics, plant propagation. We can have a assisted migration of population as to bioconserve them neatly. We can use bioindicators with the help of this AI and ML as to have a bioconservation and biomanagement systems. We can also identify invasive species with the help of AI and can protect the natural species from the invasive species. We can also have a molecular this breeding strategies and protect this biodiversity. We can manage them, we can conserve them. I wish this Professor Sarode and his colleagues a great success for this conference. Thank you.
Thank you,
Okay,
sir.
thank you so much, sir. Now, I request Dr. Dilesh Malpure, sir, to present the vote of thanks to today's inaugural chief guest, Dr. D.K. Maski, sir, Vice Chancellor, Karmavir Bhaurao Patil University, Satara.
Thank you, madam. Good morning, everyone.
सर माइक ऑन करा मालपुरे सर
Hello. Am I audible?
यस सर यस
Good morning, everyone. I am here to express gratitude towards all the gathering here. First of all, I would like to thank Honorable Vice Chancellor Dr. Maske sir for accepting our invitation and to inaugurate this one-day international e-conference as a chief guest. Thank you very much, sir, for inaugurating this e-conference and delivering a wonderful speech on biodiversity and conservation. And also, you have highlighted the application of AI in the life sciences. I am very much thankful to you, sir. I am also thankful to our principal, Honorable Dr. Sarode sir, for his welcome address and welcome address. I am also thank him for his overall support. I also extend my gratitude towards all the participants present over here. Once again, thank you everyone. Over to you, madam. Thank you.
Thank you, sir. Now it's time to keynote address. A conference of such magnitude is graced by the wisdom of exceptional minds. So we are privileged to have not just one, but four distinguished keynote speakers who will share their invaluable insights on diverse topics within the influence of life sciences, biodiversity and conservation. So I request Dr. Nilesh Malpuri sir introduce our today's keynote speaker, Professor Dr. Dipti Yakandwala ma'am, Senior Professor, Department of Botany, University of Peradeniya, Sri Lanka.
Thank you, madam. Good morning, everyone. It gives me immense pleasure to introduce today's speaker, Dr. Dipti Yakandwala, who is going to deliver a keynote speech for the one-day international e-conference. Dr. Dipti Yakandwala is presently working as a senior professor at Department of Botany, University of Peradeniya, Sri Lanka. She did her PhD in the area of molecular plant systematics and phylogenetics from University of Reading, Center of Plant Diversity and Systematics, United Kingdom. She also worked as a teaching assistant in the same university. She worked as visiting scientist at Queensland Bioscience, Prince, Australia, visiting Fulbright professor at Carnegie Museum of Natural History, Petersburg, USA. She is the fellow of Sri Lankan Association for the Advancement of Sciences and National Academy of Sciences for Sri Lanka. She is chairperson, secretary, and member of many different governmental and non-governmental bodies. To mention one of them, she is member of National Species Conservation Advisory Group formed by Government of Sri Lanka. She has been honored with many important international and national awards. Some of them to highlight are Fulbright Advanced Research and Lecturing Award, Fulbright Commission, USA, in 2009-2010, Presidential Awards for Scientific Research consecutively from 2007 to 2010 and again in the year 2024. Excellence in Research, Faculty of Sciences consecutively from 2017 to 2023. She has worked as resource person at various international and national conferences, seminar and workshops. To her credit, there are more than 86 research papers published in international reputed journals, 2 books and 6 book chapters. She had attended more than 145 seminars, conferences, and workshops at international level. Her resume is uh, very huge. It more than 35 pages. It is not possible to cover all the part of it because of time constraints. I have covered highlights from her CV. Please excuse me for curtailing your resume, madam. With this short introduction, I welcome you to this one-day international e-conference and deliver the keynote address. Thank you, everyone.
Thank you, Dr. Nilesh Malpura, for those kind words about me. And just to check, can you all hear me?
Yes, ma'am. Yes.
Okay, thank you. And first of all, I would like to thank Dr. Nilesh for inviting me for this prestigious conference, which I feel is like very important for me as a scientist from the neighboring country to join. And I'm happy to say that this is the third conference that I am joining within the to Indian conference within the past 6 months. So with that, I'll come into my today's topics. It's unveiling the regional biodiversity through molecular markers and as an effective strategy. And then to start with, I will talk about my background, in a sense that my country's background, to link this topic and the importance of my work and trying to highlight the essence of the research and the things that I'm going to talk about in the next few slides. So we as Sri Lankans, or we are situated, the island is situated at in the tip of Indian Peninsula as a small tiny island, and this biogeographic position and the varied topographic differences, and because of this we have diverse climate zones, and then this has blessed the island with 16 different floristic regions, as you can see marked on this map. And then due to these variations, we are blessed with once again 184 families, which is around 45% of the plant families of the world, and about 24 angiosperms genera. I'm sorry, can you all hear me? There's a disturbance outside.
Yeah, we could hear you, madam.
Okay, can
Please
I,
continue.
if you give me a minute, I can ask them to stop it. Oh, is it okay, my continuing?
Yes, ma'am, please.
I will continue, or is it okay continuing?
You continue, madam. Clearly, we are able to hear.
Okay, right, thank you. So then, like, if you look at the number of flowering plants we have about 3,000 odd number of flowering plants, which is not very attractive, which is not a very attractive figure if you compare the world diversity. But what I want to say, the importance, try to highlight the importance of our floral diversity through the number of endemic species that we have, which is about 20%, 28% of our flora are endemic to the country. So with that, if I move on to link we and India, as we all know, we had a shared biogeographic history, ecological past, and India and Sri Lanka were connected by land bridges during the glacial period. This facilitated the movements of plants and also animals. I'm concentrating on the plants, I'm sorry for that, through these land masses. So this connection has left a profound impact on the region's current flora, that is Indian counterpart and also Sri Lankan current flora. But later on, what happened with the divergence and the isolation of Sri Lanka during the Eocene epoch of the Paleogene period led to the evolution of a distinct plant species within the island, and also still remaining genetic connections with the Indian counterparts. So with that background, I'll come for today's theme that has been put forward for the conference, Advances in Life Sciences, Biodiversity and Conservation. So I'll just try to highlight biodiversity and conservation. And my area, I'm more or less a taxonomist, a systematist. So I will try to promote my area as to how we would try to contribute. So we know the biodiversity is a variety of life. Without knowing how many species exist and the boundaries within them, this information is given by a taxonomist. And then if this information is missing, we can't plan accurately for conservation of this biodiversity. And also, even that the shared floral diversity of Sri Lanka and India, is always we are unable to work in isolation. We need to bring up, or we need to initiate taxonomic studies involving experts from both countries, which is vital for comprehensive understanding of the regional biodiversity if we are aiming for a conservation. Because we all, the Vice Chancellor Sir just highlighted that we are, we are also contributing for one biodiversity hotspot. Western Ghats together with Sri Lanka is one biodiversity hotspot, and we know the definition of the biodiversity hotspot is that we are rich in endemics, and at the same time it's been threatened by anthropogenic activity. So we need to initiate conservation strategies to protect this biodiversity. But before we do that, we need to know what are the species and their species boundaries. So as I said, this calls for joint conservation efforts So need to collaborative research and conservation initiative between India and Sri Lanka to protect this shared biodiversity, and then cross-border projects involving common taxa, and this will bring a immense benefit for sorting the species boundaries and endemism and things like that. So at that time, when we try to talk about these conservation issues, like we need to join conservation, and then we need to share data, and also the data should be rigid data, solid data that can address these problems. So then we know the molecular markers can play, and they have been identified as a good set of data for answering species limits in cryptic species. We know what are cryptic species, and also to assess the genetic diversity, which is very important for conservation studies, even within the same species, and also which is crucial for developing effective conservation strategies. So the shared research efforts between the two countries can lead to better protection of these species and ensuring their survival for the future generations. I think it's our prime duty to conserve this, whatever the left biodiversity, for the next generations. So coming into the first half of the theme, advances in life sciences. So I would say molecular data has contributed immensely for the advances in life sciences, especially the taxonomy, and also in parallel, the data analysis methods, even AI tools, like phylogenetic methods, have provided a good platform for the taxonomists to go beyond the traditional methods and then come up with better solutions. So molecular markers, and if you look at the contribution of molecular data in taxonomic studies, we are preliminarily we are struggling to clarify the species boundaries, whether cryptic species are just one species or they could be classified as two, because if they are two, once again, conservation for conservation matters, that is very important. And also there are hybridization events, and this gene flow, this hybridization and gene flow events are once again very important. And then at that time, I need to highlight it's not only the molecular data, it's like a holistic approach we need to go. And then I have just given a slide in here. We also need data from other areas to interpret our molecular data, because if the paternal and maternal inherited gene trees are not agreeing with each other, we can't go for a combined tree. And at that time, these incongruences can be addressed through other areas, especially chromosomal studies, because we can talk about hybridization if the same taxa is appearing in two different monophyletic groups. So it's saying molecular data has provided us an advanced tool. Still, we need to use this molecular data on top of the other traditional data that's available for us. And also we can talk about the biogeographic patterns, because we are struggling to see how, or we are putting all our efforts to unveil the past biogeographic patterns, the evolutionary patterns. And at that time, we are very interested to see when our endemics got speciated, and then whether it was separated before, it happened before the separation from the mainland mass. So everywhere people are looking for answers. So all these questions can be answered through molecular data together with other data. So these are I just thought, because colors are there, I just told. So we can get data from any sort. So depending on your problem, depending on, and I said you can't work in isolation. If your group of taxa has been addressed by somebody else, maybe Indian counterpart, I will definitely go and select those gene regions and that type of data to address my one, so that we can combine the data and come up with a better resolution. So now at the top of the peak of the molecular data, like most of the European countries, and we have gone for next generation sequencing, which is a very effective method, but still we have limitations because of the funding problems. So now next few slides, I will try to highlight a few groups that I have been working, and also these are shared by Indian flora, and where I feel I would have come up with a better answer if I had a collaborative research. So I'll start with the well-known species, a medicinal plant group, the Salacia species. And then if you look at the Salacia, Sri Lanka, we have 5 species, and out of them 2 are endemic. So why I started on the Salacia is that it was Diandra that was first described, but later on there were scientists with 2 or 3 specimens. They again divided this Diandra and separated Acuminitissima, saying it's another endemic species. But we should know for conservation whether actually this is endemic or not. So because of that, we initiated this study. And if you look at the India, India has 21 species of Salacia, of which 11 are endemic to India, and also these are native species that are being shared with Sri Lankan flora. So as I said, like even how developed the molecular data is, you need to start with morphological studies, because morphology itself will take you a fair amount of, will give you a fair amount of resolution for your answer, and then whatever can't be resolved, you can go a step forward with your molecular data. So I started with the morphometric analysis, and then this is the dendrogram that came up, and if you see the 5 species, but the 2 questionable species, they come up, the number of representatives are less because they are endemics and then threatened, so they come up in a single morpho group. So here also is the same, they come up with the single morpho group. So then what we did was we took represents from this for our molecular analysis, and also incorporated data from the GenBank from already done studies, and this is the maximum likelihood tree that we came up. And if you see, very interestingly, we came up with 2 clades where the Sri Lankan Salacia got separated, and the Indian Salacia came up with another supported clade. Even though the same species were shared by the 2 countries, still they came up in 2 different places. So this question, or this race question, and also we were like interested to see what's going on. So then we tried to get more data from Indian species, and we were only able to get ITS data. So based on the ITS data and the available species or the sequences, we have built up this tree. Once again, it's a maximum likelihood tree. And if you see in this, the Sri Lankan species are separated in here. And if you take, I told you that we could get more represents from the Chinensis, Salacia chinensis, and this whole large clade is hosting this Salacia chinensis. And if you see, and this there had been a publication by Anas, Indian scientist, which I'm very grateful. We got the information from that publication, and you see that he also and his team also has raised the question of the species boundaries of this Salacia chinensis, because Kerala the Kerala accession, they nicely separate from the main group, and it's the same for us also. We came up with the same results. And if you take Sri Lankan chinensis, it grouped with the larger group in here. So Sri Lankan chinensis does form a monophyletic group with the Indian chinensis, but if you take chinensis as a whole, it's all spread across with lot of genetic diversity. Right? So here you might see there are 3 other taxa within this chinensis, and these ones like Salacia, they are labeled as Salacia reticulata because they have been labeled in the GenBank as Salacia reticulata. And if you look at the literature, it says that the origin is from Sri Lanka. So I traced back to the original paper from where these sequences came, and to see, this is a Japanese study where they have taken Salacia reticulata and extracted these biochemicals, and then as a part of their study, they have sequenced this. So I want to, like, it says, like, it doesn't give the location, but it says collected from Sri Lanka, nothing else. There's no other information or herbarium specimens. So, but I tried to get in contact with them, but it was like they didn't respond to see the collection location and to see whether this identification is correct. But I feel, and also the Indian scientists felt, that these are misidentifications. So this should be Salacia chinensis. So this opens up debate or interesting question for collaborative research. Actually, we are like with Professor Vivek Pandey, Mahe University, we are like trying to go for a collaborative study. If we succeed, we can get answers from many of these questions. And just to show a few other things related to this and the biodiversity conservation, like if you look at the records of Salacia reticulata in India, there are two nice taxonomic studies in 2021 and 2012, where it says clearly that India doesn't host Salacia reticulata, whatever the specimens and the herbarium are misidentifications. But saying that, no means that I'm trying to degrade these studies, but I'm trying to highlight the importance of taxonomists and the involvement of taxonomists to sort these things out, because the basis of any study will be the taxonomy. Right? So here, but there are two, I have taken two Indian studies in 2021 and 2013, where one come from Maharashtra and other come from the Andhra Pradesh, where they have used the medicinal, that evaluated the medicinal properties and things like that from specimens collected from Salacia reticulata. So that's where the importance lies. So once again, we need to make sure that these identifications are correct. So this comes after the taxonomic publications, and also this shows the importance of any plant-based studies where the identification is important. It's always good to collaborate with the taxonomist, so that the taxonomist will help in the proper identification of your original starting material. So now we have like come into a more or less a conclusion. This is like to be published, that taxonomic position of Salacia reticulata is going to be endemic to Sri Lanka. So with that, if I move on to another genus, Vanda, the family Orchidaceae. So this is, if you look at Vanda thwaitzii, this was described in 1898 as an endemic species by Hooker. And the interesting thing is, like, at the time of this description, they didn't have a herbarium specimen. So this is the herbarium specimens, but this specimen has gone missing. But there were two drawings which are given in here that were based on this herbarium specimen. So Hooker described this species, Vanda thwaitzii, based on these drawings which are deposited at the National Herbarium Peradeniya. And then there were no available herbaria or other clues of this occurrence. So with the time, even though this was described, later evaluations, it was decided that this species is an extinct species because nobody has collected so far. But interestingly, in 2020, there was a set of scientists, nearly after about 160 years, we were able to locate one population from the Knuckles mountain range, which is the center of the island. And then now this was a very interesting finding, and we needed to publish. This is like rediscovery after 160 years. And then we were looking for other published material, and we came across this Kuma and Kuma 1998, where they have reported Vanda thwaitzii as occurring in India. So now this is no longer going to be an endemic species. So that was nice information for us. So it said that in the Kuma and Kuma 1998 was based, they have cited that the specimens that they have examined were from the Salient Valley, collected in Kerala and few other places. So they studied this herbaria and they said this is going to be Vanda twice, even though they have been previously identified as Vanda tessellata. So with that information, we submitted our paper, and then the journal, while the review process was going on, one of the reviewers came up with the even interesting point where the reviewer pointed out or gave us the information saying that Moods in 2021 has studied this Indian Vanda twice, and then he had found it out that this is not going to be Vanda tessellata, no Vanda twice, but it's a completely different distinct species, and he named this as Vanda sateshi. So with this information, once again, the Indian flora got a new endemic, while our Vanda twice once again became endemic. So with that, again, by going to a different literature, I have come across few literature that again, the occurrence of Vanda twice is there in certain publications. So by no means I'm trying to downgrade the studies, but I'm trying to highlight the importance of collaborative work. If we can study and see with the current available data, we can solve these things. So it's another group that I am working at the present, the Impartians genus of the Balsaminaceae. And if you look at the whole world, you get around 11-20 accepted species, and Sri Lanka has 24 species, of which many are endemic. And if you look at India, the diversity is very high, about 210 species are recorded from India. So if I come to my particular species, Impartian subcaudata, this is a species that was thought to be extinct from the wild, like Vanda twice, and we were able to encounter them in the wild from 3 different populations. And then once again, when we tried to publish this, the finding of or rediscovery, we had problems. The problem was, and that is was highlighted before also in Sri Lankan studies. If you look at the revision of Sri Lankan flora, under the Impartian subcaudata, Greg Wilson is the one who did this study or the revision in the 80s, and it says that the Sri Lankan Impartian subcauda is very much similar to Indian endemic Impartian caudata, with a slight difference in the flower, sepals, and few other characters. So even in the revision, this has been cited. So when we submitted this paper of the rediscovery for the journal, one of the reviewers, I'm sure it should be an Indian reviewer, was also queried the same thing, which I'm very grateful. So it queried the same thing, saying that this could be endemic Impartius caudata, which is something again gave us a point to think twice and then redo our studies and see whether actually Sri Lankan Impartian subcaudata and Impartian caudata of India, whether they are going to be one species. If they are one, we will lose our endemicity, and then it will be synonymized under Impartius caudata. So then with that, we started the studies, collecting live material for the Sri Lankan subcaudata, and then getting information from published literature. There was a morphometric study done by one, I can't remember the publication now. And in that, we were able to extract the morphological characters, and this is not, I don't think that this is a very successful one, because we have only one coding for the Indian Impartian caudata, and these are the Indian subcaudata, Sri Lankan subcaudata. And if you see very clearly, the in Sri Lankan Impartian subcaudata are morphologically separated from the Indian caudata. So with that, if I move on to our molecular studies, where again we got information or the sequences for ITS, TERNLF, ATP beta, RBCL, they are all spacer regions, highly variable regions, and this is the maximum likelihood tree. And then if you see in here, the Indian subchordata and Sri Lankan, Indian Impartian chordata and Sri Lankan Impartian subchordata are occurring in two, even though they come in one monophyletic group, they are clearly once again separated in two different monophyletic groups, giving us evidence that they are two different species. So this is also something else that I came across in last year. There was a publication by an Indian scientist which says notes on taxonomic status and re-electrotypification of Litzia etiodaphne from the family Lauraceae. And then why is it important for us is because Litzia etiodaphne was described initially in 1886 by Hooke in Flora of British India as an endemic species to Sri Lanka. But if you look at in depth this study, this research paper, it says that Indian Litzia salinga and Litzia etiodaphne from Sri Lanka are just going to be one species, where Litzia etiodaphne was synonymized under Litzia salinga. So here, if you look at the distribution now, Litzia salinga occurs in India, Bangladesh, and Sri Lanka. And this once again gave or raised another question, I would say. This earlier, if it was two species, like we can explain these are endemic, there would be other biogeographical separations in here. But now these are distinct distribution for the species Litzia salinga. So this distinct distribution raises a question how these species got separated and survived in these very distant locations. So it could be past geological or climate change that split these populations, or even it could be long distance seed dispersal mechanisms that connected these regions at some point in the history. So this paper in 2024 now raises two important questions once again, which could be addressed with two different data sets or through collaborative research using molecular data. And this is another group that I am studying. The genus Iliocarpaceae once again has a link with the Indian counterpart of Iliocarpus. Smarty in 2001 did a revision, and this was done purely based on herbarium material. So the revision of the Iliocarpus in Sri Lanka and also the southern India. So she had done a thorough study using the molecules, the herbarium material and the morphological characters. So then she comes up with several questions and several herbarium specimens, raising whether these are going to be new species. So that's why I started on this genus, and then we were able to come up with a new species for one herbarium specimen that was raised. And we came up with several populations from three different, again, they are only confined for the high mountain regions in the center of the island. And then I just wanted to highlight another one, the species A, Iliocarpus species A, based on a herbarium specimens. It's not a Sri Lankan one, it's a South Indian one. And according to my literature surveys, I have not seen this has been worked by somebody. So this is something very good for working, and also we can do some collaborative studies and also work with the same gene regions to come up with a better answer for these questions. And then Iliocarpus amoinus, even though Smarty described this as an endemic species to Sri Lanka, there are records in the databases that this is also occurring in India. So this is something, again, if you are talking about endemics and then conserving them, and then records are there in India, because our Sri Lankan population is very, very small, confined to a very small area in the border of the protected areas. So it's always good to find another population, even though we will lose its endemicity, but then we have a genetic diversity and we can address the conservation in a better way. And this is something that I am again doing, Iliocarpus hediomus was described based on herbarium specimens by Smarty. But when we took the study and we did the thorough field collections covering the whole island, I think that's the way that we should work. Go and collect much as specimens possible and look at the morphological diversity, and on top of that we need to go for the molecular and other data, so which I did. And here this is the based on the specimens collected for this Iliocarpus idiomus, we saw that there is a morphological difference within these populations. So we did a morphometric analysis and clearly we could identify 3 different morphogroups, which is also highlighted in the principal coordinate analysis. So then next step, as I always say, I go for the molecular evidence and then I take representatives from these different morphogroups and then do the analysis, which once again, which is supported by the molecular analysis as well. So this is like finally, like we are for the publication, and then at the moment we have decided to go with variety level, but we have a small problem in there whether to go for subspecies or variety level, because there's an elevation. If you take the 3 groups, there's a demarcation by the elevation where the populations are sometimes separated. So based on that, we might decide whether it's going to be a subspecies or a variety. So then, like showing the importance of collaborative research, and then if you look at the challenges that we have, and then funding is the biggest challenge for us, but we have like opportunity to tap, and also the rules and regulations. When I say the rules and regulations, like molecular data as the data are coming is something very important, or a tool we can like address these challenges, because without exchanging the material, if we through different experts groups start working on the 2 countries separately, very well we can come up with a collaborative exchange of this material and come up with the output without like going through the hassle of rules and regulations. That will like reduce the problems that we face for going through these rules and regulations and law taking a lot of time. So I told you we have opportunities time to time. This Industry Lanka grants come, and we at the moment we have applied for Salasia, and I'm not sure whether we will succeed, but we have the opportunity to see, because even though they highlight, even though these are advertised, if you look at the areas that they highlight that they will be sponsoring, they don't have biodiversity conservation. So if you look at these different areas like food technology, plant-based medicines, biodiversity taxonomic studies is a basis for building these up. And then if there are problematic groups, before coming into their utilization, food values, medicinal properties, as I told you earlier, we need to sort them out. So it's us, the taxonomists, to convince these authorities like to incorporate this area also as a very important area for funding. I don't know how to do it, but that is the need of the hour, I would say, because if you look at the funding for taxonomic studies and biodiversity, it's like very scanty. So the other thing is like the talking about the advances, life sciences, and the advances and molecular data, and the younger generation, if you see, they always want to go for this high sophisticated data rather than concentrating on the morphological and the traditional data. So this is what is being highlighted in this, and this is the article that appeared in the Nature in 2024, which I love very much because it makes a lot of sense and gives a good point to think about, point to ponder. So if I just highlighted 2 points in here, with the rise of molecular tools, some view traditional taxonomy as outdated. mistakenly assume that molecular techniques alone can resolve taxonomic issues. This oversimplified view disregards the critical role of morphology, fieldwork, and the need for integrative approaches, so leading to the reduced interest in taxonomic and essential skills. So what is said is ultimately, because we are universities and I know that several academics are joining in here. So it's like what is said is university credit hours in taxonomy that have been reallocated for fields like attractive fields like molecular biology or biotechnology, I would say, has to be reinstated. And then we need to give a good training from this traditional morphology, chromosome data, phytochemistry, pollen data, so that even though we handle the problem from molecular data, we should have this background to interpret what's been given from the molecular data. So with that, I would like to advertise myself as a taxonomist and say the contribution for all these different areas is not like I'm telling about myself, but I want to highlight these points and build up the enthusiasm of the younger generation so that they will get proud of themselves and think more about yourself to because of the contribution as taxonomists that we can do for the biodiversity. So we are the guardians of biodiversity, and we play a vital role as taxonomists in understanding the life of Earth. And I have taken this from the Kew Gardens report on state of world's plants and fungi in 2023. And this is how they advertise the taxonomist contribution. It says plant names and locations. That's what we give. We sort everything out and give the plant name. Okay, this is the name of this plant, but we do hard work to come up to this point. Using this data, all these databases are prepared. And using this data, you define the protected areas, you prepare distribution maps, diversity estimates, extinction risk is being evaluated, and evolution studies are being done. And at the base are the taxonomist, and this taxonomist has to give up the very good output, not only based on molecular, but also we should be able to interpret this molecular data equipped with other available data as well, more like morphology and other data. So this, if I go back of the 30 years of my working as a taxonomist, I have been able to I'm sure there are few such eminent scientists joining this. I am very proud to see, I'm very respecting Professor Yadav. Professor Yadav, you're there, I saw, because we have been working with Professor Yadav very closely, communicating. And then I know he's a very great scientist, which I have a lot of respect. So he also has equally contributed. I know several others also contribute, but during my speech, I want to highlight my contribution and try to build up the enthusiasm and making yourself, the younger generation, proud of what you all are doing now. So then you are able to discover new species and also bring up with new records. What I mean by new records is, if you look at the past records, sometimes even though they have identified species as a species, sometimes that is not the species that is present. So clearing those things are also very important. So for that, I will take one example which I am working at the present, Lemnaceae. This sometimes come under Araceae, but we consider Lemnaceae as a separate family at the base of the Araceae. And we know Lemna, Wolffia, and Spirudella, and there's another genus, but we have only these three in our country. So if you look at as to why, if I say why I started on this, because the records on identification of Lemna was a point of debate, because in the red data analysis and all the flora, the records don't tally. So I thought we need to do this, because they are very tiny ones. very little morphological characters are there, but still those morphological characters are very important. So I did morphological studies with the limited data available, and also we went for molecular work as a collaborative project. So this is the molecular tree that came up with these two gene regions. And if you see, the Spirodella came or grouped with the Spirodella polyorhiza, which we didn't have a problem in identification. And if you look at Wolffia, the Sri Lankan Wolffia was identified as Wolffia rhiza, but we got a new identification because it formed a very strong monophyletic group. So these bold ones are the represents or the accessions that we have incorporated from Sri Lanka to the analysis, and the identification came as Wolffia neglecta. The most important thing came for the lemna. We had so many specimens incorporated. If you see in here, the lemna has two clades in here, and some of our Sri Lankan lemna formed a monophyletic group with Landoltia. Landoltia is a new genus that was described very recently, which is almost same as lemna, but there's a subtle difference in the morphology as well. We can see a profilum in here and few other characters. So we did from this study with Sri Lanka, we could identify a new genus to the country. And also if you look at the identification of the lemna, which we thought we have Gibba, Perpusilla or minor, they were given a completely different identity as lemna actinoptalis. So this is like based on morphology and molecular, and the molecular verification was very important because they had limited number of morphological characters. And also, if you look at the new species discovery, I take this as an example because like when we took this Laginandra genus, we have had only 6 species described, out of which 5 were endemic to our country. But once within the past 3 years, we were able to describe 6 new endemic species to the country. And if you look at the distribution, they occur in India, Sri Lanka and Bangladesh. So I'm sure if you hunt, like if the more field work is being done, I'm sure there will be new species being described to India as well. Equally, the diversity should be rich even more because the area is, compared to Sri Lanka, the area is more. So this is a very interesting group because a lot of people are interested because this has an ornamental potential as well as aquarium industry is also sometimes coming behind this aquatic plants. And also for ecological restoration, bank restoration, this is a very nice candidate. So discovering new species, how can we recognize or like look at the importance of them for the biodiversity? I would say it's a need of the hour. And why? Because if you look at the recordings or the recorded data, it says 45% of the flowering plant species are at a risk of extinction. So this brings up alarms, as if 45% is the flowering plants at risk of extinction, most of them are not known. So if these unknown plants are at risk of extinction, we don't know whether we will be able to find them out before they become extinct. If they go missing, extinct, we will not be able to know that they were there. So this is where, once again, for biodiversity conservation, taxonomies play a very important role. So then, like with the extinction risks, we come up with evaluating IUCN red listing and the conservation status. I would say the taxonomist is the best person who can give this on top of, it's not the ecologist, but the taxonomist who will give the best data, because our identifications, we are very sure. And also we go, that's why I said we go for field work and we see the habitat health, we see all the threats. So based on all those data collectively, we can contribute in a very good manner for evaluating their conservation status under the IUCN criteria B, where they take the EOO and the AOO as the main determining factor together with few other criteria. And then once again, taxonomies can contribute for building up species model, distribution models. We all know that we as signatory countries, all the signatory countries are responsible for conservation of these threatened taxa. So by preparing species distribution models, we can come up with new locations, we can contribute for restoration programs, and we can also reintroduce or introduce these for the rehabilitation or restoration programs. So I'm happy to say that we have worked with such one taxa, Indianthus variegatus, with Mr. Vishnu and Professor Vivek Pandey from Mahe University, and then we were able to come up with this collaborative paper, which was mostly done by them, and then predict the distribution and also the future distributions with the climate change, how these distributions will change, and also what would happen for the populations. And also biogeography is another area that we can we are interested. Taxonomic studies again will answer these questions, and we as taxonomists, even though molecular biologists can prepare, we as taxonomists, we can prepare the taxa which should be included in the analysis in a better way. And then based on these things, we can talk about the biogeography. I will just share one ongoing study based on Piper using rad sequence data. And if you can see in here, we have calibrated our tree, and our rad-seq based study reveals two main clades for the Sri Lankan Piper formed by the historic distribution events, dispersal events, and the diversification during the Miocene era. You can see that that's where the diversification occurs. So one consists of predominantly Sri Lankan endemics formed by the in situ speciation. We think that would have been the reason, whereas the other links the Sri Lankan taxa to the South Asian, East Asian group. I'm sorry, this is not moving here. To the Southeast Asian. So we have one endemic also in here, and Phalanx is shared with India as well. And then notably, we have some unexpected results where the affinity of Walkeri, this is an endemic with the Southeast Asian species, and also the Sri Lankan Selenicum is coming up with Indian Hymenophyllum. This is Sri Lankan Hymenophyllum. This is an native one. So the Indian Hymenophyllum we have not incorporated, but we have a sequence now. So we can answer whether this is going to be one Hymenophyllum species or whether we are going to have two different species that are different, and these are two different species for the two countries. So these interesting findings have been given out as we have come up with this interesting finding from this research, which we have opening up areas for collaborative research. So this is an ongoing one, so I will not give you details that much. And as the Vice Chancellor correctly said, alien invasive species is another threat for the biodiversity conservation, and taxonomists, this is a bonus for us when we go do the field work and we are thorough in identification. We encounter these plants. Some people will neglect it, but we as taxonomists, we will always see what is this species, and thereby we can even detect the alien invasive species in our habitats while doing our other field work. So these are few such encounters that I have been gifted by doing field work. We have been able to record Ludwig acidoids, Nymphea species, then Mica species, Echinodorus, and also the most recently the Cabomba furcata. I know South Indian, I have seen records. This is going to be a threat for the biodiversity. So this is a recent discovery for the Sri Lankan aquatic environment, which will be an important finding for biodiversity conservation and managing these invasive species. So with that, I will come into the end and then again quoting a study, a report in the Guardian newspaper based on a study from the Kew Gardens. It says botanists identify 33 global dark spots with thousands of unknown plants. So it says botanists have identified 33 dark spots. We don't know what's inside around the world, where thousands of plants are probably waiting to be discovered according to the new research. But with more than 1 lakh or 100,000 species believed to be undiscovered, the majority of which are believed to be at risk of extinction. So these dark spots, they assume that harbor lot of species that are threatened and threatened with extinction. So it is our duty for us to like shed light on these dark spots together, and that is the message that I want to share. And if you see these dark spots, I don't know why Sri Lanka is not highlighted, but India is highlighted as one of those dark spots. So as a way forward message, by harnessing the power of these molecular markers, as I showed earlier, we can transcend traditional taxonomy boundaries to reveal the hidden genetic diversity of shared flora. In doing so, India and Sri Lanka can jointly illuminate the evolutionary histories, resolve taxonomic ambiguities, and drive the conservation strategies for the rich yet underexplored biodiversity of our region. Thank you very much. And I would also like to share my collaborators without whom I wouldn't have been able to do this. Thank you very much.
Thank you, ma'am. Now session will be open for participants to ask any doubts and questions. Anybody have questions, feel free, share with ma'am. Yes, anybody have question? Participants? I'm audible? Okay, thank you, ma'am.
Okay, thank you.
Ma'am, for your valuable insights.
Thank
Now
you very
I
much.
request Dr. Jyoti sir to present the vote of thanks for today's keynote speaker.
Sorry.
Yes, ma'am.
No, sorry. I thought whether I can leave. No, I will wait for the vote of thanks. Sorry.
Good morning to everyone. On behalf of Department of Zoology, Botany and Aquaculture of SCGM College Kopargaon, I extend my deepest gratitude to our resource person of this session, Professor Dr. Dipti Alkandawala, for their informative lectures on unraveling regional biodiversity through molecular markers. Dr. Alkandawala lecture made these events with glory of knowledge. Hope participants gain deep knowledge from Dr. Alkandawala's lectures on importance of biodiversity, their importance, how to use molecular markers in biodiversity conservation, biodiversity study, describing new species, and species extinction risk assessment. Once again, thank you very much, ma'am, from the organization. Thank you.
Thank you. Thank you very much for inviting me and looking forward to like initiating collaborative research. Nice to see you, Professor Yadav, as well. So I will leave the...
I wanted to say something. Thank you very much for a very informative lecture. I was listening each and every word carefully, and it was really a feast for me. Thank you very much.
Thank you. I'm honored that you have joined. So thank you very much. So I will leave the conference. Sorry, because I have lectures after 12 o'clock.
Yes, we have.
Thank you, ma'am. Now I request Dr. Nilesh Malpuri sir to introduce and invite our today's intellectual session. First speaker,
Thank you, ma'am.
Professor Dr.
Thank you, ma'am. Everyone. It is my honor to introduce today's guest speaker for intellectual session, my guru and my mentor, because of whom what I am today, and he is none other than Honorable Professor Dr. S.R. Yadav. He doesn't need any introduction, as almost all of us know him very well. But as a protocol, I need to introduce Professor Dr. Yadav. Recently, he has been honored with as INSA honorary scientist and working at Department of Botany, Shivaji University, Kolhapur. He completed his PhD in the area of medicinal plants from University of Mumbai in 1983. Since 1977, he has held different appointments such as lecturer to professor and head at various reputed colleges from Mumbai and many different universities such as Shivaji University, Kolhapur, Goa University, Goa, University of Delhi, New Delhi. Professor Dr. Yadav has been honored with 44 national as well as international awards. Some of them to highlight are Best Teacher Award of Year 1999 by Shivaji University. Late Professor V.V. Sivarajan Gold Medal for outstanding contribution in plant systematics by Indian Association of Taxonomy of Angiosperms. Dr. R.B. Ekbote Award, R.N. Agarkar Foundation at MSCS Pune in 2008. He has been awarded Fellow of the National Academy of Sciences, India, India in 2009. He has been elected as Fellow of Maharashtra Academy of Sciences, Maharashtra in 2009. Pancham Maheshwari Medal, Indian Botanical Society 2012. UGC BSR Faculty Fellowship Award in 2016. Fellow of National Academy of Sciences in 2017. And most prestigious award, that is E.K. Janke Amal Award, given by Ministry of Environment, Forest and Climate Change in 2017. He has been honored with DSc by D.Y. Patil Deemed University in 2022. He has been the member of many different bodies. He is the life member of International Association of Plant Taxonomy IAPT since 2024. To honor Professor Dr. Yadav, a new plant species is named as Srirangia and over 18 new species are named to honor the contributions made by him. He has discovered 81 new species till date. He has guided 31 students for PhD and 11 for MPhil degree. He has organized 21 conferences, seminars, and training programs and attended over 128 conferences and delivered 92 invited lectures by at international and national conferences. To his credit, there are more than 340 research papers published at international level in international reputed journals. He has co-authored 8 books and 16 book chapters. He had attended more than 292 seminars, conferences, and workshops at national and international level. The important credit that I should not forget to mention is that he has completed research projects worth rupees 17 crores, 17 crores. And presently, he is engaged in plant exploration of Western Ghats, biosystematics, and conservation of rare plants. With this short introduction, I welcome you, sir, to this one-day international e-conference. Thank you. Over to you, sir.
Whether a slide is visible to you or no?
No, sir.
Hello? Slide? Now?
No, sir. Slide is not visible.
Is it visible now?
Yes, sir.
Yeah, right.
Yes, sir. slideshow
Yeah,
केले
yeah, I
होईल.
will make it. Is it visible now? Hello?
Yes,
Am I
sir.
audible?
Yes. Yes, sir.
Sir. It is
Yes.
visible now, sir.
Right. So, good morning to all of you. I'm thankful to Dr. Nilesh Malpure for a nice introduction. Everybody feels happy if he listens about his achievements. Anyway, that is one part of our life, but serious than that is the responsibilities of our educational and research institute in documentation, conservation, and bioprospecting of our plant resources. Just Professor Ekandwala highlighted importance of taxonomy, basic research, and that is a foundation for doing all other kinds of research, and especially with modern tools and techniques. Definitely they will add, but basic data remains basic, without which we cannot think about conservation of biodiversity. So with these words, I will go to my first slide. That is every species, is important. I show this slide many times, and it is my purpose, that is as a teacher, that how educational institute can play a major role in conservation of biodiversity. Now, every species, usually human beings, they think the species which are useful for them, like medicines, food, and other plants which are useful to human beings, they know them well and they are caring for those plants. But as a botanist or zoologist, we must care for each and every species, and all of us, we know the reasons why we should care for each and every species. Number one is every species in this world, whether it is microbe, animal, or plant, is a unique creation of nature. Unique creation of nature. Everyone is unique. Secondly, especially plants, plant species. Human beings take Nobel Prize. We say he got Nobel Prize for this and that and for DNA structure and so on. But plants are unparalleled chemists of the world. I mean, how many types of alkaloids, flavonoids they create, they synthesize, how many types of proteins, carbohydrates, and whatnot. So they are the best chemists of the world. Another thing, that every species is a witness of evolutionary history of life. All of we know every species which is around us is a witness of evolutionary history of life. Each species has phytogeographical significance. Species are not haphazardly distributed. Each species has its own habit, and they are evolved for millions of years and have phytogeographical significance. Different functions in ecosystem. Now, whenever somebody comes to me for plant identification, he says, what is this plant? And second question is, what is use of this plant? So usually he asks me what is used to human beings. But every species has its own function in an ecosystem, right? Then each species has a different range of tolerance. Some species are found widely distributed and some are restricted to a small area. We call them as endemics, endemic species. They are very habitat specific. In requirement, they are specific. So every species has its own range of tolerance and distribution. Similarly, in space, different species adjust with each other, just like a family. It is a family in which different species, including animals, microbes, and plants, they adjust, and there is a spatial relationship among different species. Then, phylogenetic relationship, they are how they are related with each other, how they are evolved, and we are trying to construct a tree of life, which is equally important. And finally, I say, the last sentence is probably more important for everyone, not only educationalist, not only botanist, zoologist, but every person of this planet. If his true capital, real capital of human beings, if it is asked, I will say it is a biological capital, which is the most important capital of any human being. That also I will explain why it is so important. And therefore, we can persist our students in of any side, arts, commerce, engineers, doctors, everyone is concerned with this subject, not only botanist or zoologist. or microbiologist. It is a subject concerned with welfare of human beings. Every branch has to be involved. So this is what I feel. And then, this slide I show, and we should show, all teachers should show to our students the origin of human beings. The hominoidea, this clade, about 20 million years ago, this got separated. Gibbons got separated. Then our orangutans got separated about 20 million years ago. 8 million years ago, one more clade got separated, gorillas. Still they are existing, And about 5 million years ago, that clade bifurcated into two clades, chimpanzees, genus Panthera, and another that is Homo sapiens, genus Homo. So chimpanzees are human beings. Our origin is considered to be about 0.2 million or 2 lakhs years before. Humans is the latecomer on this planet, and probably today it is ruling the earth. It is ruling earth, and human beings have qualities that way. This is our history, our phylogeny. Now, coming to this one, when we see science and these techniques, science has shown that about 98% human genome is of that of chimpanzees. So 98% we are chimpanzees, 2% we are human beings. With this 2%, we have done wonders on this planet, right? See that this is the latecomer, only about 2 lakhs years before we originated on this planet. The last second of 24 hours, we have originated, and then we have made progress from chimpanzees to humans, and today we are sitting in front of computers. So great developments. We talk about AI and artificial intelligence, which I don't know how it will go ahead and what will happen. Now, to know the history, see, this is the Rift Valley where we originated. We were there in that Rift Valley like chimpanzees for 1.5 lakh years. Then we started migrating. Probably in India, we came about 50,000 years before, 40,000. Then we had gone to this one, Australia. We migrated to Australia, then to Eurasia, 40,000. North America, just 20,000 years before, and South America, just 10 to 12,000 years before. Now, this is our history. Why I am showing this? Our history on this planet is only for 50,000 years. It is well proved, well known. But why I am concerned with this history? In this 50,000 years, we have changed the completely face of the earth. Entire face of the earth has been changed by human beings. Now we talk about agriculture. Just 10,000 years before, we started cultivating plants in different parts of the world, and agriculture came into dominance. It is a part of, really speaking, botany. The plants which we needed, we started cultivating, so we called it as agriculture. 10,000 years. Today, you see me, tell me, if you take survey of your own country, almost, 2/3 land we have brought under cultivation. Every piece of land in our country, we are converting into agriculture land, leaving no space for wild animals, wild plants to grow, and that way, agriculture for whom? It is only for human beings. It is not for other organisms. So in 10,000, agriculture is a major reason for loss of biodiversity all over the world initially. And now there are a number of reasons, So this is agriculture. If you see our Western Ghats, all of us, we know Western Ghat. We are from Maharashtra college. I'm in Maharashtra. And every piece, every hill is made flat, and we are growing sugarcane. So that is agriculture, economics, growth. And that way, we are harming our Western Ghat to such an extent that I don't know what will be the future of Western Ghats. Now, there are four important things which I want to tell you, on which I think every day. Water. Water is life. Jal hets jivan hai. Water is the life without which life cannot exist. Okay. Then, jal hi jivan hai. Without water, no animal, no plant, no microbes can survive in Earth. Our Mother Earth has this gift of water, which is very important for all of us. And there are four naked truths of life. Of course, sunlight is important, soil is important, minerals are important. But in between this physical and living things, there is one important part, that is plants. So water is one, which is without water, we cannot live. Number two, which always I show, gold, money, power, and so many things people are running after. But if you don't have food to eat, entire wealth is useless. So first biological capital, our food, is a biological capital, without which not only human beings, no other animal can survive. So plant has great role. Plants are the best scientists of the earth. With minimum capital of minerals, water, sunlight, and carbon dioxide, they synthesize food for entire world. Not only human beings, all animals also survive because food is available. So let us understand the importance of plants in our progress, in our history. We exist because of plants. So this is one. Second one, same thing. Hungry man will not look at the gold or dollars. He will look at food. That is the primary need of the life. Okay, basic need of the life. All other technologies, development can make your life enjoyable, but they cannot sustain our life. If you think about sustainability of human life or animal life or microbe life, we need to come to, food, which is needed. So this is one thing. Another thing which I understood, all this gold and this, I am showing examples. Food is the primary thing. And this man who has lot of love for gold, but unless he eats food, unless his stomach is full, he cannot enjoy the beauty of the gold. So finally, it comes to the food. Second one, even if you want to powerful, become gunda, do raj, okay, whatever. Even you want to become king, unless you are powerful in fighting, okay, unless you eat food, you cannot be powerful. So again, even if you want to powerful, you want a power, if you want a strength, you have to eat. So for all that, what comes, that is plants. Okay. So this is one thing of our life. Second thing, if we don't have good health, we cannot enjoy anything on this earth, whatever it is, even if it is heaven. And if you are if you have fever or if you have some disease, you cannot enjoy anything on this planet. And therefore, medicines. Food is also a medicine. We eat food in such a way that our health is maintained. In addition, if you fall sick, all medicines have come from plants. So far, plants have taken care of our health. And good health, probably health, your body is the topmost, property of your life. Good health is the topmost property. No other property is as important as our health, which is taken care of by our plants. Third is oxygen. What plants, wonderful creation of nature, just splitting hydrogen, water, giving oxygen for us, and, that way we survive. I always tell that per day human oxygen bill, it is about 21,000 rupees per day if we want to buy oxygen to live alive. This is, continuous chetana. We breathe in, breathe out, take oxygen. We never think of from where it comes. Again, it is from plant kingdom. All oxygen we have and living, living life is existing in this earth. So I told you four naked truths of life. I know all other factors are important. Water, food, medicine, and our oxygen, they exist. That's why we are doing all this progress. Even if you want to study AI and apply AI, we have to be alive. So what I feel, this is a life-sustaining science. Biology is a life-sustaining science which needs to be taken care, and plants need to be conserved. As a teacher, we all are, mostly we are teachers. Our existence, whether it is a snake, tiger, lion, frog, bird, you name the organism, their existence on this earth depends on plants. Okay, of course, animals are equally important for survival of plants, but our existence depends on plants. Now here, one story which is 373 people gave their neck to save Kazari trees, which is a story of 1730 AD. So old people were knowing importance of a tree. They know that one tree, its value is more than one's life. And that's why they gave their neck, did not allow to cut the tree. But today, people have lost their senses, and the way we are destructing our forest, I need not to talk about it. The way we are working, which will be a really disaster in coming generations. This story I am always repeating in the beginning only. Now, one leap, God Krishna, and this Satyabhama tried to wake Krishna with gold, but she failed. And then Rukmini put one leaf, it is said Tulsi, one leaf of plant that weighed heavier than God Krishna. What is the scientific message from this, which is given to entire world by our forefathers? What I have understood, that they have told, oh man, if there is something more important than God for you, it is a plant. And I told you, we exist because of plant. And probably that's why one leaf, it is an indicative of plant, is weighed heavier than God Krishna. So this is a story which we should tell to our people. Now you see, each species will weigh heavier than dollars, right? And in India, many projects are sanctioned, and they don't care for endemic plants, loss of plants. They know very well. Now see our Western Ghats. Hello? 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 Am I audible?
Yes, sir. Yes, sir. You are audible, sir.
Somebody is talking, please hint him.
No.
See, this is Western Ghat, and you see the beauty, right? One. Now you see these plants. Who is more advanced, human beings or plants? I will prove that plants are more advanced than human beings or than many other organisms. The reason is, plant can survive for thousands of years in the form of seed, without water, without air, and without food. But animal cannot survive even for 2-3 days if food, water, and air is not available. So that way they are very this one. These are our Western Ghats, and see the panoramas which give great pleasure. And this nature only, we should remain in harmony with nature. Gautam Buddha has told us, but we have forgotten it. Now these are the plants, Western Ghats from this one. And the true matrix of human life is the green world covering Mother Earth. To a man, there is nothing more essential than a plant on this planet. Really speaking, that is the truth. And then you see, this is another thing which I always think about. Whenever we get frustrated, whenever we get bored or tired, we go to the forest areas, and natural areas. And that fascinating physical, emotional, and spiritual relationship, contact with nature, makes us to become someone. And most of our Rishi Munis also were in forest areas only, they got real knowledge. So anyway, this is the Khas Plateau. You can see this plant's beauty. And then you can see this is the only Mother Earth which has life on it, right? Only Mother Earth. And we don't know whether on any other planet we have live. So far. Now I come to our educational institute. My topic is, what is the role of educational institute, research institutes in our biodiversity documentation, conservation, bioprospecting, and utilization. This is the important part. Don't underestimate the importance of our primary, secondary schools, colleges, and universities, research institute. We are playing a major role, right? So these institutes, first thing, where research is done, again, it is research institute where we bioprospect our bioresources. Primary school, we educate our children about nature and their, importance of plants, animals, their conservation, and so on. Secondary school, we do same thing. In universities, our students do research, find out new things. Colleges, we train them in various disciplines, and all these students only work for the country, and therefore country progresses. Not because of leaders, because of policymaker, country progresses. Actually, the students which we train in different colleges, universities for various courses work for country, and country progresses. Productivity of country goes up only because of trained students in different disciplines in India. So that is our contribution, right? Very rarely we think about our contributions in country developing, country making. So educational and research institute with reference to biodiversity, first thing, documentation. So I know Botanical Survey of India also, whatever students, scientists are there, they are trained by universities, by colleges. They go there, they work. So finally, we provide trained manpower to Botanical Survey, Zoological Survey for identification. They document. Many universities do documentation of biodiversity of various parts through students. So that is one documentation. Another is bioprospecting. Our research scholars, they are working on various organisms, their utilization, and various compounds or how it can be used for various purposes. All that biotechnologists are working for bioprospecting, that we are doing in universities and colleges. Then conservation. In most of the universities, we do very important service of creating awareness, not only in students, but also in society by writing articles, by talking and telling importance of biodiversity. So that is one reason we think about conservation, right? We do research also. We, take many projects for conservation, and that way conservation is also our activity. And then we come to sustainable use, how to use our bioresources sustainably. That training is also given by teachers in various colleges and universities, primary school and secondary school. So these are our functions which I have told. Now, educational and research institute are very important, and one thing, they are known as think tanks, where real thinking goes. These educational institutes think on various issues in the society with respect to biodiversity. They discuss various issues. Today's conference is also a think tank, what needs to be done. Title of the conference itself is telling biodiversity, its conservation and utilization. This, where we are training students, participants, telling them what is the importance. So train and introduce biodiversity to students, train in identification and documentation of biodiversity, create awareness, research scholars create scientific information through their research projects and contributions of students and research scholars in documentation, bioprospecting, sustainable use, and conservation of biodiversity is a recommendable job. This, which I feel from my heart, a very important service is given by educational institutes. So creation of trained manpower for the task of biodiversity documentation and conservation is done by universities, colleges, schools, and so on, right? So this is one I should tell. And now you see, what is your strength? Our strength, education. I am giving you data of 2017, right? See, Central University is 54. Each university has more than 500, 400 acres of land. State universities, 411. Deemed universities, 123. Private universities, 282. Total universities, 875 universities. Colleges, 33,900 in India. Standalone universities, 7,154. Students enrolled at graduate level, 34 million students are enrolled every year for graduation. See the strength with educational institutes. They can do really wonders if we train them accordingly. Okay, so here training, creating awareness, all these things are done by universities, okay, and colleges. Now, why and how students are of prime importance in documentation, bioprospecting, and conservation of biodiversity. Number one, student strength, their enthusiasm. Students with great enthusiasm, great strength, and great potential, great energy, they are with you. Understanding of students and love for nature. We educate them. Scientific knowledge about importance of biodiversity. It is also we convey to our students, teach them strong wish to care for Mother Earth. Today, every young man is caring for his earth, his Mother Earth. You can see that was not in my time. Knowledge of plant species, their identification, propagation, multiplication, they have today up-to-date information of technological innovations they are using for biodiversity study and conservation. Innovative minds, enormous energy, enthusiasm, and strong desire to work. These are the students' qualities, and we are going to guide them in future, especially with reference to biodiversity. I have great hope and confidence that students, research scholars, and teaching community are playing and will play a crucial role in creation of awareness in society, documentation, bioprospecting, and conservation of biodiversity. If you see the past, in last 20 years, our colleges and universities have created this awareness in society and people talk about. Now, our country, one of the richest country, I need not to say much about it. I work in angiosperms. All other groups are equally important. Animals are equally important. So there are 21,849 species of angiosperms alone recorded from India. Remember, 10% of total world species occur in India. Others, gymnosperms, bryophyte, algae, fungi, all these are there. I will not go to in those figures, but total plant kingdom is represented by 54,700 species, about 700 species. Now, this Western Ghat. Now, this is a Western Ghat. All of us, we are concerned with, although it is an international conference, but especially people from peninsular India are directly concerned with Western Ghat. It plays major role in hydrological cycle, climate of the peninsular India, and biodiversity. It is a treasure house of biodiversity of our country. One of the 8 hottest hotspot of the world, this Western Ghat. It is playing great role. All perennial rivers originate in Western Ghats, and entire peninsular India agriculture is irrigated by these rivers. So our agriculture is dependent, climate is dependent, we have rich biodiversity, and that way Western Ghats are important, and many things probably people know well what is happening with Western Ghats. Now, in Western Ghat, so far 7,400 species are recorded, of which 5,588 are indigenous, 2,250 are endemic to India, found in Western Ghat. Exclusively 1,273 are endemic to Western Ghats. All these endemic species, many people don't know. Exotic and naturalized species, 376. Cultivated and planted species, 1,436. 38. So this is the data which you should know. That's why I have shown. Karst plateau I have shown already. See this Karst plateau. There we want to exploit bauxite for aluminium. You know, our botanist only, zoologist only have opposed this mining in Western Ghats and stopped some of the minings, not all. So see the biodiversity they have shown to the people. This is the biodiversity. Once it is lost, it will be lost forever. We cannot bring it back. And many mining programs have been stopped. So this is our contribution. Okay. See this Karst plateau. See this diversity, you know. Whatever you say, human beings know real pleasure of human life comes from plants. See the flowers, their beauty, animals, plants. You know, many people are very fond of so many animals, charismatic animals, birds, butterflies, tiger, lion, you know, crores of rupees they spend to see this, you know, how many wildlife sanctuaries people visit. So see this is also a one gift from nature, right? Diversity of flowers I am showing, right? See this diversity. And you know, nature is very complicated. Just Anonasi flowers, if you see the diversity in flowers alone. Can you understand why this is a diversification? Why so many types of flowers, habits are developed? All that is really probably it is those who work in nature, field work, which is a probably basis for every progress in biology. They only can understand why it is so, right? The plant-animal interactions which we study. See these flowers of Western Ghats, and I don't have words to explain their beauty. See this acanthus, and many times I say that we take plants, animals for granted. For granted, we don't care. We don't think about them. But if you see closely, really, you will fall in love of nature, love of plants, love of animals, right? Many people, they are passionate. They work on various groups of plants and animals and try to know, not only for, you know, publishing paper. It is something beyond that. Paper is a biodata, just a biodata. I don't care for it. But those who are really interested to see beauty of the nature, creation of the nature, you can see this diversity, right? Diversity of Barlerians. See this diversity of Arissimas, right? Diversity of Arissimas. Then see this. So just I showed you some flowers only. Leaves, stem, habit, all those are equally fascinating things to know. But flowers are more fascinating because of their colors, right? So these are the plants. Now we come to the important point, crucial role of education and research institute. First thing I told you, already this slide is repeating, documentation. Sorry, here we can play important role, educational institutes, agriculture institute, horticultural institute, where domestication of our wild plants as ornamentals. You know, I have taken survey. None of the Indian plant is in horticultural trade today. Why? We have not domesticated, we have not improved, so we have not utilized for domestication. There are n number of plants which we can domesticate and bioprospect them as ornamentals. Then screening for phytochemicals. 2,200 species are endemic to Western Ghats. Phytochemically, if they are screened, definitely you are bound to get some of the biochemicals, active biochemical from those plants, right? Similarly, new resources from plant diversity. One number of new resources we can get. And today, in time of climate change, we need to improve our crops, or that wild relatives of cultivated plants are of great significance, where botanists, zoologists, and microbiologists can play important role, right? So one, domestication as an ornamental. Just giving lectures is one part, and actually doing it is another part. Practical approach is important. in Shivaji University lead botanical garden, we have brought about 25 barlerias out of 30, and they are grown as ornamentals. You can see none of them was used as ornamental. We have shown that these are the barlerias which can be easily propagated and planted in gardens, in landscaping gardening, or as a border plant, as a hedge plant, as a age plant, and we have shown. So domestication of ornamental plants, ornamental plants. Then wild relatives of cultivated plants, you can see this, Vigna aconitifolia, Vigna mungo, Vigna radiatus. These are 3 crops, moong, udid, matki. All these are important pulses of our country, and they have many wild relatives which only taxonomists, field workers know where they occur and how they grow. So that kind of research needs to be done is done by universities and research institute like BSI, ZSI, NBRI, NBPGRI. Right. Now another C, grasses. Of course, all of you know, but many times we don't understand importance. Grasses is a most important bioresource of the earth. We take grasses for grasslands for granted. Really speaking, it is very simple. If you want to save your charismatic animals like your leopard, tiger, and other organisms, animals, then you need to have grasses in your forest area. Then only herbivores, then only tiger reserve. So this is very simple. 80% of human requirement come from grasses. Although it is a truth, naked truth, nobody takes care of, nobody does research on grasses. We do research on only, new drugs, something new, but grasses is the most important bioresource. Somebody should do. Educational institute, not only they do this one, documentation, but academic research, which is the basis. Today, everybody wants applied research. Who will do academic research, which is equally important? And these educational institutes, education, one of our important function is educate society, educate people. Another is research. Third one is academic. Then conservation, awareness, creating awareness in society, utilization, all these functions are done by educationalists. So, for example, academics, I am showing this academic. This is a plant which we described, Trithuria concanensis, and this plant was kept near to Eriocolacea and Gramini. Now, with molecular studies, it has been shown that it is a basal family which comes near to Nymphaceae. It is a basal angiosperm, which is an academic contribution. Probably it has no any application from which we cannot make money, but this is a very important contribution made by Shivaji University Botany Department. So this is about academic, one example. Now, education, number of students. Now we have established a lead botanical garden in Shivaji University. Such gardens can be established in every university, every college. Young students want to come, want to know animals, want to know plants. In Shivaji University, exhibition of insects was done, and so many children came to see these exhibitions, because today children in cities, they have not seen ant also or mungla also, so they don't know. Such exhibitions and then creating awareness in society, educating them is a very important. Now this lead botanical garden is playing important role. All our universities should develop such gardens, which will also help in ex situ conservation. So number of students, teachers, research scholars, training in lead botanical garden, more than 10,000 students visit lead botanical garden of Shivaji University and get education. So you can understand the importance of educational institute in creating awareness and education, nature education. So that is what I want to say. And here you can see universities and colleges have strength of talented and hardworking, enthusiastic and passionate students. They can do wonders in, biodiversity conservation. Now, this is training programs. You can see these training programs. Today also it is a conference. It is also one part of it. Introduction of lead botanical garden, then practical sessions, training them in different groups, identification, then theory sessions, field training at Tilari, making them to how to do field work. Then only we document biodiversity. No, all these are the things which are done by college teachers, university teachers, and research institutes. So this way we play important role, right? Now forest department is one who is supposed to take care of our forests. Now all of you know, all teachers and this one, those who UPSC, MPSC exam they clear and they get job as a DFO, RFO, how many of them are able to identify plants and animals of our forests? Only few charismatic animals, only few important trees. Today, they are supposed to know each and every plant species, animal species, so that they can take care of those species and conserve them. Are they equipped? Answer is no. Again, educational institute have to train them, and we conduct workshops, training programs for forest officers, and that way try to help them in knowing biodiversity of their forest so that they can take care and they can conserve and they can utilize. So all these rare, endangered and threatened rare species, what to conserve, why to conserve, how to conserve, where to conserve, all these questions have to be answered by only educationalists. And these are the monotypic endemic tree genera of Western Ghats. If we can bring in botanical gardens, our student will do research, bioprospecting, do tissue culture, whatever they want, and they will come to know. But unless we bring them in botanical gardens, they will not be able to reach those plants or they will not know these plants. So this is one example you can see. And Delphinium malbaricum, you can see, then Sagria, Loripulia, all these plants, Garcinia indica, Garcinia talboti, all these are our bioresources. Plesioneron indicum, Mamiya suriga. I will tell you a story. Only 1 month before there was a conference on Mamiya suriga, Surangi. This tree, which is almost endemic to India only. Some part of Ceylon also, some few plants grow. But Mamiya suringa, there is one area where this Mamiya suriga grows and the people take care of this. In 4 villages, I have been told that flower buds of this plant, 4 crore rupees income they get. It is utilized. And this awareness is also created by a college. They conducted conference and made it popular plant, brought it in the limelight. How we play important role. Thakursia montana, you can see Hydnocarpus, Hopia ponga, Vatica chinensis, whatever I am showing, most of those plants have been brought in Shivaji University, lead botanical gardens, so that people can come, see, and do some research, right? These are endemic plants, Irinocarpus, see this, important Disoxylum gotteadora, then Disoxylum malbaricum. This is an endangered plant. Okay, only in sacred groves we get. And they have 22 glycosides have been detected in this highly medicinal Pictosporum. So most of the day. Now this is one ecosystem, Myristica swamp, only 2.5 acres in Maharashtra, and it can be destroyed anytime. These are unique plants. You can see this Myristica magnifica, Myristica magnifica. This is Gymnacanthera canary. Why I am showing? These are the things which are known only by educationalists, researchers, universities. No minister, no common person, no leader, they know these things. Only we educational institutes, research institute do study and document. See these palms of our country. Okay. because of time constraint, I will be going fast. Now see this Corypha umbraculifera, right? See this palm. Now these palms, I will say a lot of things on it, but why not to bring these palms in our garden? Now these palms are beautiful palms. Can we bring them in gardens? Now Shivaji University has brought exotic plus indigenous 100, more than 110 palm species in garden. So that is also contribution. You can see this, right? Now this is one example. Our own trees in Western Ghats or in Himalaya or in Eastern Ghats, why not to bring in gardens? Okay, why not to bring along the roadsides? Why you want only Spathodia, that Tabobia, and all those exotic trees all along the roads? Even we people grow our trees along the roadsides, our indigenous, we can conserve a lot of biodiversity and we can make germplasm bank, okay, field gene banks all along the railway tracks and highways and so on. For highways, crores of rupees are spent, okay, but for plantation of proper trees, nobody thinks. Probably it is our failure. We teachers have failed, we researchers have failed to convince our politicians what needs to be done. Now this is Harpullia arborea, which grows in wild. These fruits hang on the tree for almost 2 months and it looks beautiful. Why not to grow such a trees as a ornamentals in our garden? Just some example. Polygarna arnalciana, Polygarna grama, Piliostigma, all these are rare endemic plants. Okay. This is Comifera berry. Okay. This is Hopia. Then your Hopia, all these plants which we have grown in our garden. See this impatience. See, so many impatience are there. What I want to tell here, educational institutes are doing a lot of work, but probably we don't have, for education, minimum care is given. Minimum funding is given for research, biological research, minimum funding is given. And that is probably we are suffering. All this is a wealth of country which needs to be utilized and taken care of and conserved. So this is our wealth. Okay. So I am showing you some wealth through this Carissa enormis. See the fruit, it is small mango. Bioprospecting of this fruit for achar and for various purposes can be done by our student and develop as a fruit crop, Bomansia, right? So my Shivaji University Botany Department has described more than 81 species, and that is a great contribution. You can say 81 new species from our Maharashtra. All these are 81 new species, and only one example I will give you. Crinum malbaricum is described from Kerala. We collected it from Kerala, and see this. This is that plant. Our students, they are wonderful guys. They can do wonders for us. So this is that crinum, and they are looking at plants and telling me, see sir, this is a beautiful plant, and it is a one unique plant which we described from India. Importance is this one, that it has longest leaves. So it is a very important discovery. 15 feet deep grows, 5 centimeter per day. Then it is restricted to 2.5 square kilometers. Again, Educational Institute only has given this data. So it needs priority for conservation. It is allied to Thyanum. It is a tetraploid, which is a academic research, and it has highest galanthamine content, which is applied research. So on one species, so many aspects come. And this is brought to light by our college teachers, research guides, and research institute. I am saying you why we are so important today. And that is the thing. Another thing, our one teacher described some 5 new species from a small pond. And you see this beauty of plants. I need not to explain. Wonderful things. What we can learn from these things. right? This is one of the most beautiful flower described by my laboratory. It is Coriandra elegans. You should see it in nature, right? This is a long list of endangered plants of our Maharashtra. So some domestication we have done. Some restoration of species is also done by the Educational Institute, Research Institute with libraries. And this Ubardia we have restored at 100 places. Now there are more than 10,000 plants and it is removed from critically endangered category by IUCN. These are the Ceropegias of our Maharashtra. We need to conserve, and this knowledge is only with you, botanists, zoologists, microbiologists. We need all of them, and you can see this hardening, tissue culture, making these samples and going back and planting in nature with students. So dear, this is already done restoration of some species. This is Songanta, Abitulan ranadi, which is critically endangered. One girl made many seeds through artificial pollination, raised saplings, and replanted in nature with forest. So these are the things which I show. Seed collection is a simple experiment. In Shivaji University, every year we collect seeds and then we give it to Malis. They raise saplings in thousands. And then, this is one university is doing, one department. And we have more than 800 universities in India. If each university does like this, most of the plants we can bring under cultivation, we can bring, and that way we can conserve our biodiversity. So you can see this nursery and these palms in Shivaji University, plantation programs. This is our soldiers, our tigers, which work, and see this restoration of species, creating awareness programs. Biodiversity is the only basis, all these functions are done by us. So biodiversity, information and knowledge creation by Education and Research Institute is the basis. Endemics, threatened plant species, methods for conservation of bioresources, declaration of megadiverse countries, utilization of bioresources, leaders, laws, and policymakers, all they depend on this biodiversity data, which is created by universities and colleges. That's why our role is great role. Okay. Conclusion, biodiversity is the basis of life. Protecting Mother Earth and welfare of mankind is our, education has that great role. Documentation, bioprospecting, utilization, conservation. In every aspect, we are probably most important people. Role of educational and educational institute, teaching, training, and research has a great significance. That is what message I want to give you. Although I can give very important research which I have done, that is not important. Now, important is how we can document, how we can bioprospect our biodiversity. That is probably very important. And educational institutes are backbone of the country. Country's backbone is education. Unfortunately, this backbone is becoming weak and weak with new policies. So, plant diversity, a biological capital of the country, region, and man. It is the only true capital of man. And this slide I have shown, this also I have shown. Plants, of course, animals are equally important. Without animals, no pollination, no fruits, but still plants are more important as friends, doctors, artists, designers, engineers, economists, technicians, and teachers. You name things, everything we get from plants. And most important question, that present generation, how they are going to hand over this Mother Earth to coming generation. Whether you want to utilize all the resources and make our earth denuded and then hand over to our children, or keep it intact as much as possible and give it to students, our coming generation, is a major question. Only educationalists think about this question. No other political party or politician think about that. How we are going to hand over our mother to coming generation. Unfortunate part of present day, right? So anyway, I always say all teachers, they have a noble job to do on this planet. They have noble thing they are doing, and their strength is their students. My students, my strength. Thank you very much. And we are working for this coming generation.
Thank you. Thank you so much, sir. Now session will be open for participants to ask any doubts and questions. Anybody have questions? Feel free to ask the sir. Yes, anybody have question? Audience,
Hello.
participants.
Yes.
Hello.
Hello.
Yes, sir.
Sir, my question is to conserve endangered plants and to collect their seeds from forest. What kind of permission we will require?
We have to take permission from our forest department for collection. Okay, NBRI, this National Biodiversity Authority. Only if we want to send outside, then we need permission. Otherwise, we don't need. And from forest department, state department, we have to take permissions for collection of these seeds of rare, endangered, or any plant from protected areas.
So, sir, if the plant is not from protected area and it is an endangered plant, can we collect and raise it?
Definitely, that is the only way we can save it, right? See, collecting seeds and raising saplings, what is wrong with it? I mean, whatever, you can openly discuss with me, because this question is asked, why we should bring, it is growing there. No, it is endangered. And under this situation, when it will become extinct, nobody knows. But if we bring these seeds, grow them in different botanical gardens, private gardens, roadside, it will come under human care. And definitely we can save that genome. Okay, but if it is in wild, and if we don't do anything, definitely if something happens, it is going to become extinct. And once it becomes extinct, you cannot bring it back.
Yes, sir. Thank you, sir.
Thank you, sir.
Okay.
Once again, I'm asking you, anybody have questions? Thank you, sir.
No questions.
Thank you so much, sir. It's my privilege to propose the vote of thanks on this session lecture given by Professor Dr. S.R. Yadav, sir. I extend my heartfelt gratitude to our distinguished guest speakers, Dr. S.R. Yadav, sir, for sharing the valuable insights and expertise with us today. Sir, your knowledge and experience have provided us with a wealth of information and inspiration. We are truly honored to have you join us, sir. Sir has explained very well the various implication species, academic plants, the origin of agriculture last 10 years ago, and Jal Jeevan Hai, and sir also showing very nice diversity of flowering plants. No doubt, sir, this will enlighten our participants to learn many valuable things, and after this lecture, they will be able to explain and explore subject concept in a better way. Once again, thank you so much, sir. Before moving ahead, I request all the participants kindly mute your mic and the camera for minutes to taking the photograph. Okay, thank you, participants.
Let us now transition to our second enlightening intellectual session, where fresh perspectives and thought-provoking ideas await us. With great pride and immense enthusiasm, I extend a heartfelt invitation to our second keynote speaker, the eminent and visionary Dr. Shakti Kumar Singh, Scientist D at Ministry of Environment, Forest and Climate Change, New Delhi. He has renowned personality in the domain of life sciences, Dr. Singh has made significant strides in research and development. His tireless effort and passion for science have illuminated new paths in biological exploration. Today, we are honored to have him among us to share his vast reservoir of knowledge and expertise. To give a formal introduction and shed more light on Dr. Singh's exponential academic and professional journey, I would request R.A. Meshram sir to do the honor. Over to you, sir.
Thank you, ma'am. Good afternoon, everyone. Joining us from India. Good afternoon, everyone. Joining us from India and other parts of the world. We just finished our first session, and now we are starting the We just finished our first session. And now we are starting the second session of this international online conference. It is my great pleasure to welcome our next respected speaker, Dr. Shakti Kumar Singh. Dr. Singh is working as a scientist at the Ministry of Environment, Forest and Climate Change in New Delhi. He completed his PhD in zoology from the well-known Banaras Hindu University. He has more than 15 years of experience in the study of insect and biodiversity. His work focuses on fruit flies, sand flies, and diseases like Kala Azar. He has also worked on the insect identification, rearing techniques, and the method to control disease-causing insects. Earlier, he worked as a senior entomologist at the Kala Azar Medical Research Center in Bihar and as a research associate at the Indian Agriculture Research Institute, New Delhi. He also served as a head of department at the National Museum of Natural History under the same ministry. Dr. Singh is a well-known expert in this field of insect and the conservation, and we are very happy to have him with us today. So without further delay, let us warmly welcome Dr. Shakti Kumar Singh for his keynote address. Let us all welcome Dr. Shakti Kumar Singh for his session.
Thank you so much, Ankita and Meshram sir. Am I audible?
Yes, sir. Yes, sir.
Thank you, sir, for your articulate introduction to Dr. Singh. Your words have magnificently captured his remarkable journey and significant contributions to the field. As we continue our quest for deeper understanding in world of life sciences, let us prepare ourselves to inspired and enlightened by words of Dr. Singh. Let us extend a warm and respectful welcome to him as he shares his profound knowledge and thought-provoking insights. With eager anticipation, we now hand over session to you, sir.
Thank you so much once again. Whether my slide is visible or not, can you please confirm?
Yes, sir. Yes, sir. Visible.
Yes, sir.
Okay. So thank you so much once again for inviting me to deliver lecture on the importance of biology in relation to the vector-borne disease and also horticultural crop pest. Before start with my lectures, I would like to just inform you that my complete talk is based in two halves. One half is on infectious disease, that is through sandfly, and the second half is a pest species on pest species belongs to a family Tephritid fruit fly families. That is, most people confuse that fruit fly families are only Drosophilidae, but there is also another fruit fly which is called a true fruit fly family, that is Tephritid family. All these are pests of major horticultural crops. So whether my slide is changing or not, please confirm.
Yes, sir, it's changing.
Okay.
You can make it a full screen, please. Make it a full screen. Right.
Okay. Okay, so just I want to focus in my talk that basic research is the most important research. Wherever there is a vector-borne disease is concerned, and even in horticultural pest species is concerned. So first, I would like to start with the sandfly that is belong to a family called Psychodidae, and there are total 5 subfamilies, out of which 2 families which are included in the public health and veterinary importance. Mainly they are belongs to the subfamily Psychodidae and Phlebotomini. So there is a very much confusion about these 2 subfamilies. Most of you people have even seen these insects in your bathroom, and it is a very small insects, but that is belong to Psychodidae, which are of veterinary importance, and the Phlebotomini are mainly for the disease perspective view. These 2 subfamily have their different habitat. Psychodidae mostly breed in aquatic and semi-aquatic habitat. This includes seashores, margins of stream, pond, rice field, etc. And Phlebotomini mostly in humid terrestrial habitat. So from this slide, you can visualize there is a picture of a mosquito, that is a female mosquito, and the second picture is of sandfly. If you compare the size, you will come to know that the sandflies is almost 10 times smaller or very much smaller compared to the mosquito. And their feeding behavior is also very much different. When mosquito feed, it makes sounds before feeding. Before feeding, it makes sounds, buzzing sounds. But sandfly are called silent feeder. Sandfly are called silent feeder. They don't even make any noise when they are in the periphery. And if you compare with their morphology, you can see the Phlebotomini are more hairy, wings, delicate, long, and cylinder leg. Wings have parallel venation, cross venations when present restricted to the base of wings. There are various morphological characters based on it. Many species are differentiated, but I will only focus on a species called Phlebotomus argentipus, which is a known vector for visceral leishmaniasis called Kala Azar. There are almost 800 species worldwide in this group, out of which 80s have been implicated as a vector of human disease. And they are mainly in 3 groups of genera, in New World also and in Old World also. New World means in American countries, and Old World is like ours and Oriental, Asian, and Indian subcontinent. So these are the method of collection of these insects. For this, mainly a CDC light trap on the extreme right side. This is a CDC light trap, which is a battery operated trap. It has been used for collection of these flies. And you can see the habitat where these flies are found. Actually, these are also neglected insects. Like the disease itself is a neglected tropical disease, which means it is found only in the tropical regions of the countries and mainly confined to the Bihar, Bhutan, in India, Bihar, Calcutta, Bhubaneswar, and these areas, and even in the part of Uttar Pradesh, but in Nepal also, Bhutan, and Myanmar. So in household, you can see here with the use of mouth aspirator, which is having HEPA filters. This mouth aspirator with HEPA filters mainly used for collections, and sometimes because the nature of the fly is very peculiar, they like to reside in the darker corner of the house and mud house. And this Kala Azar is also known as a socio-economic disease. Why it is called socio-economic disease? Because it is related to the health of the humans and the socio-economic conditions like sanitations and that, because that provide a conducive environment for their growth and multiplications. So when you see the biology, this particular insect is a homometabolous. That is, it is having all stages of eggs, larvae, pupa, and adults. They required humid but not wet conditions, and at least 70 to 80% relative humidities, and nearly, because these temperate regions provide such conditions for their better life cycles. Only female takes the blood, and male feeds on the plants, nectars, and juices. There are two main genera which are mainly belongs to this Phlebotomini group, that is Phlebotomus and Lidzomia. Lidzomia is in the New World countries, that in the American regions, and Phlebotomus is in the Indian subcontinent and Ethiopia also. When you see the life history, you come to know that these are terrestrial in all life forms, in striking contrast to the other nematocerans, where it is requires wet conditions, like in mosquitoes. Nematocerans flies means mainly, as an example, you can say mosquitoes. The generation times measures in weeks, months, rather than days, in case of these phlebotomites. So they are causative agents, or they are vectors of various viral diseases like Chandipura virus diseases, bacterial diseases, that is Bartonellosis, which is not in India, or very rare, and protozoan, that is Leishmaniasis. They also transmit some other viruses and other diseases like sandfly fevers, group of viruses, Toxona and Punatotoro virus in South American central part, Chandipur in India and West Africa. Phlebotomus species is suspected vector in India, Senegal, and also in Papatassi also. Papatassi, even other species, that is a Phlebotomus papatassi, which causes the Papatassi fevers. There are a form of Leishmaniasis, which is called cutaneous Leishmaniasis, diffuse cutaneous Leishmaniasis, and mucocutaneous Leishmaniasis, and visceral Leishmaniasis. The visceral Leishmaniasis, called Kala Azar, or sometimes it is also known as Dam-Dam disease, is mainly because of protozoan Leishmania donovani. Leishmania infantum is other pathogen or protozoans which is caused the visceral Leishmaniasis in Mediterranean basins, and Leishmania chagasi is in South Africa. So before we start with any epidemiological studies, we need to know about the vector triad or epidemiological triad. This triad consists of three main groups, that is parasites, vectors, and host. And to control the disease or protect ourselves from infections, we must block the interaction between at least two elements in the epidemiological triad. That is, even we can cut parasite vector, vector host, and parasite host. I will get into the details what it resulted. So there are factors which contribute to the Leishmania transmissions. The parasite vector interaction resulting in infections of the sandfly midgut, subsequently development to an infective stage parasites, then vector host interactions, that is a reservoir interactions, resulted in the transmission of disease, which is one of the most important that I will focus later on, because our study is mainly on the transmissions that we have done in the Bihar area. And the parasite host interactions resulted in the disease where treatment is required. Now, as I told, the visceral leishmaniasis is an infectious disease, and it is a neglected tropical disease caused by obligate intracellular protozoan parasite Leishmania donovan. And there are two modes of transmission of this disease. One is anthroponotic, that is, humans are reservoirs, and the second one is zoonotic, means canines and dogs and other animals are reservoirs. This is the basic cycles of transmissions of through anthroponotic mode and the zoonotic mode. I will mainly discuss about these two modes, but zoonotic mode is not properly known in India. They are mainly in the Leishmania, other species, not Donovani, that is Leishmania infantum, and in the American countries and other countries. Now, this is a cycle. I will not explain the cycle, but later on I will go into the details. But briefly, I would like to mention here that these paraprotozoans have two forms, that is amastigote forms and promastigote forms. The promastigote forms develop in the insects, that is in the vector, and the amastigote is in the human, where the disease occurs. So now I was telling about this species, Phlebotomus argentipus. This is a male on the right side, and female fully engrossed with the abdomen is full with the blood. The species name itself suggests that they are not strong feeder. Phlebotomus means the vein cutter. Phlebotomus is a Latin word. It means vein cutter. These flies only cut the vein and feed. They don't have a strong proboscis like mosquito where they suck the blood. So you may be heard about that there is a post called phlebotomist, where that particular person have the responsibility to draw the blood whenever you go for any blood parameter observations. So similarly, this is Phlebotomus species cut the vein, and they are not having this strong proboscis to suck the blood. This is a setup for development. Before begin with this, I would like to state here that in 2014, we as a team with BHU and NIH have formulated a project that is mainly to know the transmission mode. As I earlier told that in Indian subcontinent, transmission of visceral leishmaniasis or Kala-Azar is through anthroponotic mode. It means humans are reservoir, but there were no confirmatory experimental proof which properly depict or revealed that which groups of the disease persons or endemic regions are transmitting the disease, or how transmissions occurs. Means the reservoir is a place from where these flies pick the parasites, and that is a protozoans. And it is very much important. It is very much important to find out the reservoir for a total control of the disease. So for that, a study called xenodiagnosis, that is human xenodiagnosis study, was planned. The study xenodiagnosis means Diagnosis outside, that means you have, for any kind of a disease, when you go to the doctor, he will take or suggest to check your blood parameters and other parameters to know, based on symptom, to know what disease you have. But in xenodiagnosis, we are not using any human symptoms which is causing the disease, but we are detecting the flies with the parasites, for which first there is a basic research plan that is of rearing of a specific pathogen-free colony of Phlebotomus argentipus. So for the development of a specific pathogen-free insect colony of Phlebotomus argentipus, this is the setup required, the BOD, for rearing of isolines. I will get into the details one by one. So to develop the a colony of Phlebotomus argentipus, we know, as I already earlier told you, we need to collect these flies and live flies from the field and take this fly into the laboratory. The laboratory setup should have a proper animal room. That animal room, because this is, I mentioned earlier that this is a blood feeding insects. It required blood meal for completion of its life cycle. To provide the blood meals, there are various other way also, but in our experiment, we use mice, and that mice have been checked properly for any other diseases. And then we collected the flies from the field, and with the help of these mouth aspirators, we sorted out and put into the jar. From jar, each with the help of a vial, a single female, gravid female, have been collected and put into the isoline. And these isolines rear separately, for which we required a food that is mainly prepared from the beat of this rabbit, and then it is properly fermented and then grind it properly, autoclave, and then use. This is a main feed for the larvae, because the immature stages requires such feeds. So what we did, this is an schematic diagram where we have initiated the colony, and that first part was the isoline rearing, where what we have done, the wild-caught sandflies from the light traps were transferred to a holding cage and sorted by species and sex, because when you collect from the field, it may be having some other species also, because in field there are also other species like Sargentomia. These are also sandfly species, but they are not known vectors of any disease. But Phlebotomus argentipus is separated. It is identified and kept in the vials. You can see here, in a single vial, single female, gravid female, have been kept. And then from this vial, the blood-fed, unfed, gravid argentipus were set up as an isoline to produce F1 generation. From wild-caught to F1 generations, identification was confirmed upon the death of the females. Before, means we do not throw away. After the fly die, sometimes it lay eggs, sometimes it won't lay. If somebody have kept vial with not a gravid female or not a blood-fed female, then it will not lay egg. If it is a gravid female, then it will definitely lay eggs. And then after few days, the flies died. Then we collect the egg, separate the egg, and identify the flies, because we have to confirm the flies which we are using for development of the colony should be Phlebotomus argentipus. Then we mixed, after in the F1 generation, after the eggs hatch and larvae develop to the second instar, isoline were consolidated into 125 ml pots, and 10 to 20 isolines vials per pot that accumulated and keep in the others. The content of 125 ml pots were consolidated into 500 ml pot, 5 smaller pot to each larger pot. Immature stages were reared to F1 generation. Then the F1 adults emerge into the cage, because after emergence we have to separate it with the help of mouth aspirator, keep in the cage, and then transfer into the feeding chamber. That is another cage which is of bigger size, and you can see in the diagram, a schematic diagram here, a rabbit, it has been restrained. For restraining, we have developed design with the help of a wooden block. We have developed a restrainer where these rabbit have been kept for feeding, for the blood meal, because this fly, to complete the life cycle, they required blood meals. So before keeping this rabbit into the chamber, what we did, we shaved the belly of the rabbit so that it will provide proper space for feeding of the blood. Because as I told you that these flies are not strong, having not a strong proboscis, and they only feed blood through cutting the veins. So you can see if you, I'm not able to zoom these pictures, but if you zoom these pictures, so mostly the belly area and the ear area, the fly will feed. So F1 adult progenies of wild-caught flies were released into the mating. Before that, I have missed, because we have to release these flies into the bigger chamber or a bigger cage for matings. And then 72-hour post-emergence, the females were offered blood meal on a restrained rabbit. The blood-fed females were kept for 24 hours and set for oviposition pots for egg laying and progeny rear to adult. Then the F2 generation adults and subsequent generation progeny please excuse me. Hello. Yes. Yes. Regarding what? Actually, I'm in a conference. I will let you know after. Can you call me around 4 o'clock? Okay. Sorry for the interruptions. So I was talking about the rearing or from isolines to mass rearing and expansion of the colony. So likewise, we have reared from F wild to F1 generation to F2 generations, and then because it is a very much known fact that before doing any xenodiagnosis study, a colony should be developed, and that should be a specific pathogen-free colony. It means the flies which is being used for the development or for the xenodiagnosis study should not have any Leishmania parasites in their body. Then only we can know whether these flies are getting the protozoans or the parasites from the cases, for which we have divided 3 groups of populations. One group is called the active VL cases. We were having hospitals where people with Kalas are enrolled, and these people have been enrolled for the xenodiagnosis study also. Then the second group is the treated and the active group, the PKDL people and the asymptomatic groups. The asymptomatic, this word you may be heard before also. They don't have the symptoms of the disease, but their serological data indicates that in due course of time, few asymptomatic people progress into the disease. So we categorize these three groups of people for the xenodiagnosis study. But before doing the xenodiagnosis study, what we did, we have certified our colony that our colony is a specific pathogen-free colony through screening. That is viral screening, that we already know that Chandipura virus and sandfly fever virus is there is known in Phlebotomus argentipus. Then routinely we check the blood meal of host, rabbit for leishmaniasis parasites, and through the molecular method also. So these pictures, you can see the developmental stages of the sandfly Phlebotomus argentipus. Here you can see egg, the first instar larvae. It is having total 4 instar. The final instar stop feeding. You can see in the third instar, the gut is filled with the whatever feed, because it is almost translucent. Whatever the feed they take, it can be visible. And then it pupate, and after pupations, the adult emerges. That is a female adult, and it will hardly take 35 to 40. For details, this work has already been published in Journal of Medical Entomology in 2017. For details of these initiations, isoline rearing, and expansion of the colony, you can visit and see the article. So, as I told earlier, that human xenodiagnosis, that for human xenodiagnosis, what we did is, the step I will first let you know. Approximately 30 to 40 females argentipus, Phlebotomus argentipus used, and that is of 3 to 4 day old, because the mature flies is required for proper feeding, and it is starved, at least 12-hour starved, to feed on the ear and axillary area of the subject animal for at least 30 minutes under local environmental conditions. Because they are very fragile and adapted to laboratory conditions, that is at 27 degree and 18, flies were used for xenodiagnosis in the field and transported between the labs and the field site to customize styrofoam container and ice pack cotton. The flies detected after 60 hours post-meal and examined under microscope for the presence of Leishmania promastigotes, or through DNA extraction from the fed flies. This is how we do our experiment. We have done already, this experiment has already been conducted, and that work is also published. So we have, this is the second, you can see here, this is a feeding chamber. That feeding chamber, in this case, it is one of our collaborators who was interested, that he was very much sure that after certification of fly, that fly is completely free of the pathogens that cause the visceral leishmaniasis. So he has tried on his own hand, and you can see here the flies are feeding. So then after this, the flies were examined under microscopes, and it has been found that the active VL cases are the main reservoirs which has been established for the first times. This is one of its kind study, and that work has been published in Lancet Microbe. And here, at least we have studied almost 287 individuals with a varying clinical status, like as I already told, asymptomatic infection, active VL, and PKDL. PKDL is called is a further complication of this visceral leishmaniasis, that is called post-dermal Kala-Azar disease. Because the name, I forgot to tell you that visceral means visceral leishmaniasis. That name itself indicated that it infects our visceral organ of the body, and mostly when these parasites enter into the body, they attack on the macrophages. You can see macrophages are play major role in engulfing of any pathogen introduced into the body, and they use these macrophages as a house, and then finally they go inside into the spleen and reside there. So it is that the disease, the symptoms of disease is very much related to like other most fever disease, even in fact dengue, common fevers, and some other malaria and chikungunya disease like that. But it is not easy to detect. It is not easy to detect these parasites even with the expert doctors, because it required for 100% confirmation, it requires splenic puncture. It means to get this parasite from, to know the parasite from the infected persons, we have to take the spleen out, which is a very complicated procedure, because sometimes if you not properly puncture this spleen, then that patient may die also. So it is a very complicated way to diagnose. So for that, and because there is a government plan to eliminate, earlier there was a plan of to eliminate by 17, which has been extended, and we are on the verge of eliminations by 30. But for this, we have to have an integrated approach, that is of vector control also, that of we have to know about the reservoir also. So the key finding of this study was that the infections in the endemic area are contributed primarily by active visceral leishmaniasis or active cases, and the PKDL. PKDL is when sometimes the patient remains untreated, or because of any reasons their doses or whatever, mostly liposomal amphotericin B. Nowadays, this is a common drug, liposomal amphotericin B, which is being used to treat visceral leishmaniasis. So if that patient is not completely treated, or the parasite hide from the or escape from our immune system, then these parasites grow and comes in on the skins, and that skin becomes a very much deteriorated. And there are other many types of PKDLs that I will not going into the details, but please excuse me. But these two cases are mostly responsible for the reservoirs that has now been established, because earlier there were no experimental proof of this. The proportion of participants of active visceral leishmaniasis is in fact at least 55% of sandflies when accessed microscopy, and 78% when accessed with quantitative PCRs from the pooled midgut. This study was also done to evaluate the presence and absence of Donovan, Leishmania donovani, only during the early stage of the parasitic growth and the development of the vector. But we do not know which, if any, of the positive flies would go to carry the mature transmissible infections. There is another aspects of this, that Whether these parasites have been picked by the sandfly through blood or through skin is an open question. Still, there are a lot of studies that is still going on these aspects, that whether these parasites are being picked by the fly through the skins or blood, or you can say blood versus skin. So skin biopsy, but in our study, we do not obtain any skin biopsy from the patient of active VL, and we do not know whether the parasite number in these tissues is correlated with the positive xenodiagnosis or not. Also, treatment of the patient with active VLs with a single dose of liposomal amphotericin B, a drug which is used for treatment of these, resulted in low and undetectable level of the parasite in the blood after 30 days and greatly reduced the infectiousness of the patient. Whereas none of the flies were positive by microscopy. That I have already told, especially the asymptomatic conditions, none of the flies were positive by microscopy. But pooled of the flies that have been fed on 7 treated patients were positive of quantitative PCR. Patient with nodular PKDL, this is one of the post-dermal Kala-Azar disease, where nodule is formed, were more infectious to sandfly than the patient with macular, where there is no nodules, both in the proportion of flies that we are infected in the number of parasite transmitted. We did not observe any single infection as accessed by either microscopy or quantitative PCRs in 6,219 sandflies that acquires blood meal from 184 asymptomatic conditions. It means we do not found any microscopic positive parasite result in case of asymptomatic conditions, but we do found that the PKDL and the active VL cases are the major reservoirs. While conducting this study, one of our collaborator was also having an opinion that as in other countries, but except Indian subcontinent, there is a zoonotic transmission of the disease. So we have also conducted the xenodiagnosis study in animals. I'm not going into the much details, but this is a process of animal xenodiagnosis, wherein why we have concluded this, because when we were collecting these flies from the wild and we do the blood meal analysis, our work suggests that the blood fed by the flies have some source blood from the dogs, from animals like cows, buffaloes, and goats. So for that, we have studied, even in rodent also, we have studied the xenodiagnosis work in animals, including dogs. This is a process. This is the process where the xenodiagnosis in dogs that has been done. It is there in our publications, and this study has also been published. This is in emerging infectious disease. The study conducted was to investigate the Donovani infections in dog and the potential of such infections to be transmitted back to the flies. Means our aim was to know whether these dogs are also playing a role of reservoir or not. And we found the potential of the dogs, but we have not found in other. And it is somehow suggest a possible links between the canine infections and the human exposure. Also, one of the study, while doing this xenodiagnosis work, we come across the finding that For control of these insects, mainly in Bihar and in Indian subcontinent, DDT was mainly used, and that is indoor residual spraying was done to control the insects. But recently in 2015, synthetic pyrethroids have been used for control of the disease, for control of the vector, for the control of the vectors. But we know that DDT and pyrethroids both have the same target site, and there are several studies which state or mention that DDT, these vectors develop resistance against DDT. So our aim was to investigate. Whether the new synthetic pyrethroids that is being used in the IRAs, and mainly that alpha cypermethrin is being used, are causing any resistance or not. For that also the studies was planned, and also the study was also to investigate and to rear another colony of specific pathogen free colony of a specific resistant colony of that particular insecticide. So this study, because after introduction there were no report which says or reveals the effective diagnostic dose and time to assess the susceptibility of sandfly. We know that resistance is a natural process, and it is a evolutionary process. In the natural selections, the good gene is being selected. But when you use some added pressure like insecticides or some other chemicals to control these vectors, it is acting or as another selective pressure to select the resistance genes, and which may create a problem in controlling the vectors. So no validation diagnostic dose available for the sandfly till 2022. Henceforth, quantify the lethal concentrations value and determine diagnostic dose and time using laboratory reared Flavotimer argentipus were planned, and to assess the susceptibility status of the wild-caught sandflies to synthetic pyrethroid alpha cypermethrin. So what we did is we checked the LC50, LC90, and LC95 for alpha cypermethrin, chloropyrifos, malathion, DDT, and delta methrin. These studies this is a process which is by which we have collected the sandfly from the different locations. In our cases, we use 10 populations. Then we have an uncoated bottle, then that insecticide coated bottle, recorded for knockdown and transfer into the knockdown fly and transfer into the cages. Wait for 24 hours and record the mortality. That is a process which it is also being published in, and the detail you may get from there. So the diagnostic dose was established, that is 3 micrograms per ml. Specifically, I have not given for other insecticide, only given for that alpha cypermethrin. And this work has been published in Close Neglected Tropical Disease. This table is for the diagnostic dose, knockdown, diagnostic time to knockdown, and diagnostic dose of mortality for all these 5 insecticides. Then we have studied the resistance status of wild-caught flies. among 10 population that I already told you, and from this we have come to know that there are some population like here in Anandpur Kharani, that is a populations where it is clearly resistant is showing, and the rest of the population does not show any resistance, and they are susceptible to the insecticides. Secondly, this has been done in the F1 generation also, and in F1 generations we found out that when we have done this study in wild caught, that Anandpur Kharani is showing resistance, but here in 3 villages, that is Pandey, Hirapur and Hamidpur, they are resistance, showing the resistance. This study has also been published in Parasites and Vectors, so you can get the details from here. Whether my time is there or hello.
5 minutes left, sir.
Am I audible or not?
Yes, sir. You are
Yes, audible, sir.
So the second study was for biology of the fruit flies. Shall I start? Shall I start?
Sir, excuse me, sir.
Yes, tell me.
Sir, actually, we are a bit short of time. So I request you to wrap up.
Okay. So as I already told you that my second part of the study was on the fruit fly, and mainly the Tephrotidae family. So this family is well-known pest of many horticultural crops, and they are very much economically important, and there are many species which cause serious damage to the crop pest, like Bactrocera dorsalis, Bactrocera zonata, Bactrocera cucurbiti, and Bactrocera tau. These are some common species. I am naming them. These species are most serious pest of the horticultural crops, especially mango crops and guava and many horticultural fruits. So here also I would like to suggest ask our students and researchers to start their work and to focus more on the basic research, because see, by using a simple insect rearing technique, you can get more and more information and more advanced studies you can plan simply by rearing the insects. You can study the biodiversity, you can study the insecticide resistance study, you can study taxonomy, you can study the ecology, you can study resistance, how insects are getting resistance, and also you can focus on some new aspects of the morphology. As we know that insects are being only identified at the adult stages. There are, except few groups, there mostly insects are being identified based on adults. So by rearing these insects under laboratory conditions, and studying the biology, you can plan your studies. You can have more and more information on their ecology and behavior and the conservation of the insects. So here I would like to conclude or end up my talk. Thank you so much. And if any questions, I would like to answer these questions.
Thank you, sir, for sharing such insightful and thought-provoking reflections. Your words have truly added depth and value to this session. We now open the floor for interactive question and answer sessions. If you have any questions or doubt you would like to share, please feel free to proceed.
Hello, Dr. Shakti. Can I?
Hello, sir.
Yes,
Hello, sir.
sure.
नमस्कार, sir.
Namaskar. कैसे हो?
बढ़िया, sir. बहुत अच्छा, sir.
It was nice lecture, but I have only one curiosity to ask you.
Yes, sir. Please.
Can I ask?
Yes, sir. Please, sir. Please.
After leishmaniasis, post leishmaniasis, PKDL, so notes are formed, and notes you have been studied the notes also. So had you found any parasites, that is the leishmaniasis, in the notes, and what are the, if they are found, what are the percentage in notes?
Sir, this has already been established that the parasites are being picked from these nodes, even without node also, because PKDL have at least 2 to 3 form, that is the nodular form, macular form. The nodular form is sometimes is the more aggravated stages where the parasitic load is much more there. And from our experiment also that has been published in Lancet Microbe, and the percentage is above 78. The picking of this, the parasite from this node is about 78%. That has already been done in our studies. And even in active VL cases also, we have found that around 55% of these. But still, because there is a lot of research is required to be done also, because these parasites, how these parasites are being picked up by the fly is again a complicated interaction, because they are into the visceral part. That as I told you that visceral leishmaniasis, this parasite reside in the very visceral organ of our body like spleen. But when the fly is biting on the human, there are several factors which indicate, but still studies is required to be done, that how these parasites are getting the indications that yes, fly is biting on my hand or my legs, and this parasite has to be picked up so that they can go into the insect vector to multiply them, because the multiplication of the parasite and then transmission of the disease mainly because of the vector, because vector in the vector we know that insect is having open circulatory systems. There are various mode available where in open circulatory system, whenever the parasite picked by the vector, sometimes it through that parasitic movement, sometimes it flush out the parasite also. So that parasite should have to be established inside the vector, and then it should after establishing, it should multiply. Sometimes it is there that the parasite has been picked by the vector, but the disease they are not able to multiply inside the vector. So based on that, there are also several type of I think 2-3 types of like sorrow leishmaniasis. That leishmaniasis in lizard. That is a primitive where the method of multiplications is in a hind gut. This parasite multiply in the hind gut. But in case of visceral leishmaniasis, it multiplies in the mid gut epithelium. When the peritropic membrane is formed after the blood meal, they will because they enter into the body in amestic goat form. They don't have the flagella, but inside only they form this in the meiosis, they multiply, then form the flagella, then go into the stromal valve, and then they make a plug so that when they actually when they take the second blood, means they regurgitate. Actually, they regurgitate the parasite into the human, and then that causes the disease. So yes, it is established that nodular PKDL is the reservoir, or flies are picking the parasite from this.
Okay, thank you. Nice lecture delivered by you.
Thank you so much, sir.
And
let
the other have.
I would like to take this opportunity that sir is my guru, and and because of sir effort, he only called me to give the lecture. And right now I am not involved in this work. My major work is in administration and some other part. But this, whatever the result I have revealed here or presented in my talk, is my earlier studies that has been done from 2014 to 2018. So thank you so much, sir, for your blessings and your encouragement. I am still finding some place so that I can continue my research work.
Yeah, definitely. Yes.
And also, I would like to thank Vilas sir. I was not knowing. I got his called after the sir introduction. He was my senior in when I was doing my MSc. After very long time, I just interacted him. And thank you so much for inviting me for this lecture.
Thank you, sir.
Thank you, sir, for addressing the question with such clarity and precision. Now I invite A.B. Gore ma'am to come forward and express our collective gratitude for this insightful session. Her words of appreciation will surely reflect the spirit of thankfulness we all share.
Thank you, ma'am. Good afternoon, everyone. Am I audible?
Yes, ma'am. Yes, ma'am.
Good afternoon, everyone. My heartfelt gratitude to Dr. Shakti Kumar Singh, sir, our esteemed chairperson, for guiding this insightful and informative session. Sir shares the knowledge about vector-borne disease. This lecture is very informative, sir. And sir told us what is the difference between mosquito and sandfly, how we can identify the sandfly as compared to the mosquito, and insecticide resistance study also sir told. After that, sir told how xenodiagnosis done in dogs. Thank you very much, sir. This is a very informative lecture. I also thankful to Deepak Barsagde, sir, Dr. Farmer, Head Department of Zoology, RTM, Nagpur University, Nagpur, for joining us, sir. Thank you, sir. Thank you very much.
Okay, all the best.
Thank you, sir.
Thank you, Shakti. We will meet again. I will come to Delhi and meet.
Thank you.
Yeah.
So may I leave this conference?
Yes.
Thank you so much, sir. Thank you.
Thank you, ma'am, for graciously delivering appreciation. Before moving forward, I request all the participants to please switch on your cameras and mute your mic for a minute to take a snapshot. I also request all the participants to fill the feedback form provided on the WhatsApp group. I inform to participants that stay tuned for the further information on the WhatsApp group. Now I announce a short break to refresh and recharge. Please be back and ready as our next session will begin promptly at 2 p.m. Stay tuned for more enriching discussions ahead.
We'll be back at 2 p.m.
You can join with the same link again.